ਤੁਸੀਂ ਪਨੀਤ ਕੌਰ ਮਤਲਬ ਤੁਸੀਂ ਇਹ ਇਕੱਲੇ ਹੋਰ ਤਾਂ ਨਹੀਂ ਕੋਈ ਬੋਲਣ ਵਾਲਾ ਹੈਗਾ ਮੈਂ ਸਟਾਰਟਿੰਗ ਤੋਂ ਮੈਂ ਕਿਹਨੂੰ ਗੋਸਟ ਬਣਾਵਾਂ ਸੋਨੂੰ ਗੋਸਟ ਬਣਾਵਾਂ ਬੱਬੂ ਆਪਣੀ ਕਾਨਫਰੰਸ ਵਾਸਤੇ ਕਹੀ ਜਾਂਦੀ ਹੈ ਵੀ ਉਹਨਾਂ ਨੂੰ ਰਜਿਸਟਰ ਕਰਵਾ ਦਿਓ ਤੁਸੀਂ ਕਰਾਣਾ ਕਿ ਨਹੀਂ ਕਰਾਣਾ ਫਿਰ ਅਸੀਂ ਸੋਚਾਂਗੇ ਬੱਬੂ ਬਾਜਵਾ ਸੱਚੀ ਮੈਡਮ ਨੇ ਸਵੇਰੇ ਕੀ ਪਾਇਆ ਸੀਗਾ ਵਨ ਮੋਰ ਕਰਦੀ
Hello, Pandit. Hello, Pandit. Am I
Hello,
audible?
Hanji ma'am. It's fine, ma'am. Perfectly fine. It's fine, ma'am. Perfectly fine. Hello. Good morning, ma'am.
Hello, good morning.
Good morning, ma'am.
Sonia.
hello Anji ma'am good morning
Good morning.
और ma'am how are you
Fine. So actually we are arranging my presentation and shifting in the other computer.
okay ma'am okay take your time
Just a few minutes.
and yes ma'am आगे ना ये कभी ना कुछ बच्चे हमने
Actually there was a problem in my old desktop. That's why they have shifted new one.
okay ma'am okay
But it's taking time.
okay ma'am
Otherwise I was ready to give through my mobile only.
नहीं
But
मैम मैम अभी गुरप्रीत मैम की ना थोड़ी सी है 10 मिनट्स की स्पीच 10-15 मिनट लगेंगे अभी
हाँ हाँ हो जाएगा ना okay. You just see 3 people are standing with me. I hope that my presentation will be useful for you people.
Yes ma'am of course share screen oh test Ma'am, it's visible. It's visible to us.
आप ही आ रहे हो
हाँ
बाकी you can run.
Please run. मैम रन करके देखना स्लाइड्स रन होती है.
रन रन करो रन मैडम
नेट टेस्ट करना पड़ेगा ना.
हाँ जी मैम हो रही है
हो रही है. एरर भी कर सकते हो आप.
हाँ जी.
बाकी ना ये यहाँ से करने के लिए ये बैक है ये नेक्स्ट है ये आप करते जाइएगा यहाँ से. और ठीक है.
Yes ma'am, it's running.
तो व्हाट एल्स रिक्वायर्ड जो मेरा म्यूट करना नहीं अभी नहीं.
अगर देखो म्यूट करना हो वैसे ही लाते ये म्यूट हो गया.
थैंक यू बच्चों. हेलो मैडम. Is it everyone is online or they are available there only?
नहीं मैम अभी 5 to 10 minutes लगेंगे मम्मी. मैडम अब ज्वाइन करेंगे ना स्टूडेंट्स.
No problem. So should I shut down this or low कर देते हैं? I'll make it.
हाँ जी मैम. हाँ जी.
बस ओके. इसको डाउन कर दिया बस मैंने. या ज्वाइन करना है. मैडम कह रही थी अभी टाइम लगेगा.
कोई बात नहीं.
तो यहाँ से दिखाओगे ना तुम. यहाँ ना जूम पे
यहाँ से आप
बंद करो.
हाँ आप माइक
मैम हेलो मैम. हेलो
मैम.
मैम स्टॉप शेयर कर दो स्क्रीन अभी. एक्चुअली हम यहाँ से ना कुछ सेटिंग्स करेंगे फिर उसके बाद 15 मिनट्स के बाद है लेक्चर.
ओके शेयरिंग स्टॉप कर दो.
ओके.
यस
स्टॉप शेयर नहीं शेयर स्टॉप शेयर करना स्क्रीन को. अरे यार इसको सॉल्व करना है.
आपको तो लगा
Good morning, Dr. Anu. I am already there.
Good morning, ma'am.
Good morning.
Shall we start, ma'am?
आप देख लीजिए
ठीक है मैम, ठीक है, सब ने ज्वाइन कर लिया मैम. हाँ जी, हाँ जी, ठीक है मैम. So very good morning. I'm Dr. Paneet from GHG Khalsa College of Pharmacy, Guru Sudha, and I welcome everyone on the third day of Faculty Development Program on exploring multidisciplinary avenues in pharmaceutical education and research. In the last 2 days, eminent speakers from various reputed universities and institutes have shared their respective research. And moving ahead with the same mandate, we have planned 3 technical sessions for our participants today. For today's session, we have Dr. Sunita Garg, Emeritus Scientist, CSIR Nescare, New Delhi, Dr. Vinod Tiwari from IIT Varanasi, and Dr. Ranveer Singh Dhaiya from Central University of Punjab, Bathinda. We also cherish the auspicious presence of Dr. Gurpreet Kaur, co-convener APTI Women Forum Punjab State Branch and Professor and Dean Faculty of Medicine, Department of Pharmaceutical Sciences and Drug Research, Punjabi University, Patiala, as our guest of honor for the day. I warmly welcome our guest of honor and speakers for the day. A very good morning. To begin with today's proceeding, I welcome Dr. Anu Jindal, Professor in Pharmaceutics, GHG Khalsa College of Pharmacy, Guru Sudha, to deliver the welcome address. Over to you, ma'am.
Thank you, Dr. Paneet. Good morning, everyone. I'm Dr. Anu Jindal, and I'm going to present today's welcome address. On behalf of management, principal, and staff of GHG Khalsa College of Pharmacy, Guru Sudha, I welcome you all on the third day of 2-week faculty development program organized by this college. Today, we are going to have formal address by guest of honor and 3 technical sessions by eminent speakers. I welcome all the speakers. First of all, I welcome today's guest of honor, very respected Dr. Gurpreet Kaur, Professor, Punjabi University, Patiala. Thank you so much, dear ma'am, for accepting our invitation to deliver formal address and for adding glory to this event. I welcome an eminent figure, Dr. Sunita Garg, Scientist, Nescare, CSIR, New Delhi. She will be first speaker of the day for technical session 1. Dr. Sunita Garg will highlight innovative and practical aspects on the topic plant collection and identification for research and development. Thank you so much, dear ma'am, for your consent for an informative session. I welcome Dr. Vinod Tiwari, Assistant Professor, BHU, Varanasi. He will be speaker for technical session 2. He will enlighten the audience with knowledge on the topic pain, a blessing or curse, a scientific perspective. I welcome you, sir, and appreciate your efforts for sparing your valuable time and for your consent to share your knowledge with the faculty. I welcome Dr. Ranveer Singh Dhaiya, Associate Professor at Central University of Punjab, Bathinda. He will be speaker for the day for technical session 3. He will share his knowledge on very dynamic topic, alternatives to animals in pharmaceutical research. I welcome you, sir, and highly appreciate you for your consent and for selecting such a versatile topic, very useful in research. Once again, I welcome all the speakers of third day of this FDP. I also welcome all the participants of this FDP, including faculty and research scholars from various institutes. I'm sure all participants will be enlightened with knowledge on latest trends in pharmaceutical education and research. So without any delay, let's start with further proceeding. Thank you so much, ladies and gentlemen. Over to you, Dr. Paneet. Thank you.
Thank you so much, ma'am. So we have today with us Dr. Gurpreet Kaur. Dr. Gurpreet is Professor and Dean, Faculty of Medicine, Department of Pharmaceutical Sciences and Drug Research, Punjabi University, Patiala. She is a Director, Center for Product Development through Novel Technology Intervention. She is an experienced teacher and researcher with more than 60 publications in international peer-reviewed journals. She has handled government-funded projects worth 1.4 crore. She is a life member of EPTI, IPA, Punjab Science Academy, SPUR. She has been a member of Board of Studies and Selection Committees of various universities: Kurukshetra University, UIPS Punjab University, Chandigarh, Guru Kashi University, Talwandi Sabo, Shulini University, Solan, UPES, Dehradun. She is a member Project Evaluation Committee for DST Serve, AICT Travel Grant, and AICT Evaluation Committee member for inspection in various colleges and universities across India. She has guided 4 PhDs and many postgraduate students. I welcome you, ma'am. It has been our privilege to have you today as our guest of honor. I request you to kindly address our participants with your wise words. Over to you, ma'am.
Thank you, Dr. Parneet. Am I audible?
Yes, ma'am, you are.
Okay. So, respected Dr. Satvinder Kaur, Principal, GHG Khalsa College of Pharmacy and convener of APTI Punjab State Branch, Dr. Anu, Professor, GHG Khalsa College, the distinguished speakers of today's technical sessions, Dr. Sunita Garg, Dr. Vinod Tiwari, and Dr. Randhir Dahiya, and dear participants, a very good morning. First of all, I'd like to congratulate GHG Khalsa College of Pharmacy for organizing this FTP on exploring multidisciplinary avenues in pharmaceutical education and research. Now, the field of pharmaceutical education and research is changing rapidly. It's a very dynamic field. And after COVID, we have seen the immense role of pharmacists in the healthcare system. So the emergence of new medications, the drug therapies, drug repurposing, the rapidly growing field of alternative medicine, this has fueled, like anything, the growth of our profession. And this is also very true that we can now not work in isolation. The role of pharmacists in patient care, in development of policies, standards, research on new moieties, the formulation of dosage form, this is expanding like anything. In addition to this, the emergence of telepharmacy during COVID times, the 3D printing, the emergence of personalized medicine, our role is expanding like anything, we are moving like anything. Now, the mission of our education in pharmacy is to provide the students with a curriculum that will enable them to possess the knowledge as well as the skills to the drug-related, to cater to the drug-related fields, drug-related needs of the patient in the society. Furthermore, we all know AI is expanding like anything. The emergence of artificial medicine is there, virtual health. So all these have driven an exponential change in our healthcare system. So to keep abreast of all these developments, we as teachers need to update our knowledge. And thus, this is where the role of FTP comes. And the aim of these FTPs is to enhance the teaching and research skills of the people, of the faculty, of the staff, to make them aware what recent is going in the field of pharmacy. So I'm very happy to see that the organizers of this FTP have tried to rope in the speakers from varied fields so that all the areas of pharmacies are covered. We have eminent speakers from industry, from institutes of eminence, who will share their experience regarding the latest and the current developments in the field of education and research. And I hope that all the participants will be benefited from this.
Sorry to interrupt you, ma'am. Ma'am, आप mute mode पे हैं. Please
I think I was unmute. Somebody muted me. By mistake, I think.
I don't know, ma'am. It might be some technical issue.
Right. So, so I would like to congratulate the APTI Punjab State Branch, especially the Women Forum, for organizing this first event, and hope that they'll continue to do so in the coming years. My best wishes for the successful completion of this FDP, and I would like to congratulate the organizers for providing me with this opportunity for sharing my thoughts with you. My best of luck to you all. Thank you all.
Thank you so much, ma'am, for your kind, wise words and very encouraging words. We hope we will be able to fulfill the mandate with which we started this particular FTP. Thank you so much, ma'am. So it's time to felicitate our worthy guest of honor. Ma'am, please accept this small token of love from our side.
Thank you, Dr. Deol. I'd like to share that I started my career with this college. So
I know, ma'am.
Yeah, so there's a special bonding. I've got a special bonding with this college,
Yes,
and
ma'am.
Dr. Satvinder. It's like a home too.
We do share the same feelings with you, ma'am.
Thank you.
Thank you so much, ma'am.
Okay.
So let's start with our technical sessions for the day. For our first technical session, we have with us Dr. Sunita Garg. A brief introduction about ma'am. Dr. Sunita Garg did her MSc in Botany from Meerut University in 1977 and PhD from Botanical Survey of India, Dehradun. She has worked with Botanical Survey of India, Howrah, Kolkata, NBRI, FRI, and other herbaria, universities, colleges, and institutes in India for taxonomic studies. She has delivered invited talks and lectures at various forums in colleges, universities, institutes in India, and Third World Congress on yoga, Ayurveda, and traditional medicines held in 1989 at Italy. Dr. Garg joined Nescare in 1980, and she has a vast experience of 42 years at CSIR Nescare. She has worked as plant taxonomist, science writer, editor, and communicator. She joined Nescare as, right now she's working with Nescare as CSIR Emeritus Scientist, and she joined that in 2016 after her superannuation and worked on a project entitled Digitalization of Morphological Key Characteristics of Crude Herbal Drugs. She has also worked as Chief Scientist Head, Wealth of India, and Encyclopedia of Indian Raw Materials and Industrial Products, Raw Material Herbarium and Museum, Delhi, RHMD. Sales and Marketing, Editor of Indian Journal of Natural Products and Resources, Repository at CSIR Nescare, New Delhi. Raw Material Herbarium and Museum, Delhi, RHMD, is established as an adjunct to Wealth of India, houses herbarium sheets and crude drugs, animals, and mineral species of economic importance. All recognized herbaria have their acronym as allotted by International Association of Plant Taxonomy, New York, Botanical Gardens, USA. And RHMD is indexed in the journal named Index Herbariorum, which is published by this reputed institute. RHMD is frequently visited by students, entrepreneurs, and researchers from all disciplines for getting authentication certificates. Online open access RHMD e-bulletin was also started by Dr. Garg in 2017. It is the first bulletin of its kind containing photographs of authenticated samples along with other details. Ma'am has a very long, experienced, and a very rewarding career, and she has won various accolades on the occasion of International Women's Day in 2019, she was honored by Ministry of Small and Medium Enterprises, Government of India, Export Promotion Council, with countries partner like Nigeria, Russia, Ghana, Rwanda, for exemplary contribution in environment category. On the occasion of CSIR Foundation Day, Dr. Garg was honored by Director CSIR Nescare for academic interactions with school students and other visitors at RHMT. She is currently involved in authentication of fresh plants and dried crude drugs. She's sharing knowledge on health hazards from poisonous properties of various plants and their parts, collection of crude drugs like dried bark, seeds, leaves, etc., with their scientific botanical names versus common name for strengthening skill development and startup entrepreneurship potential. She's providing exposure to utilize traditional knowledge for the protection of life along with identity of plants and parts and toxicity reported in some of them. As she is facilitating as expert for the visits to RHMT and encourage people of various sectors in collection of genuine and interesting samples for classroom activities, development of pharmaceuticals, and exploring MSME activities out of their work experience. So today we are very grateful to ma'am that she has agreed to deliver a talk, and ma'am is going to speak on plant collection and identification for research and development. We are so honored to have you, ma'am.
You. I think my intro has become bigger than my presentation.
No, ma'am, your profile is very, very, very encouraging, ma'am.
I think really I'm thinking thankful to you that you are so nice and not leaving a single line adding to my.
Ma'am, there was nothing to leave, ma'am. There was nothing to leave.
Okay, thank you. Anyways, I hope that my presentation will be useful further for identification as well as you people bringing closer to the botany and taxonomy other than the pharmacy, because I don't know each single word of pharmacy studies, pharmaceutical studies, and most of the samples we are receiving from pharmacy colleges also. And I am happy to share with you that on 17th and 23rd, Anandpuria Pharmacy College, they are coming to visit our institute as well as our campus. They said, ma'am, we have to take the students for a field visit. So what is better than this place? And because you will be demonstrating as well as, because we have a very big campus here and more than 135 trees are here, and including herbs, I do tell them. So they every year they come, and other colleges are also coming for visit to her. You are also welcome to take your, bring your students
we
here.
will definitely
So in
come, ma'am. This is the first step to our association. Yes.
And moreover, I should not forget to say that long back I visited this your college as a speaker, and in Ludhiana. There was some Sardar ji, I forget his name, as a head of the division. And then he called me for a speech because my relatives are also there in Ludhiana. So I came readily delivered a talk long back, I think. I came. So now it's high time now. I'll start my speech. Is it comfortable? Okay. Manoj, link करना था ना तुमको? Madam,
Presentation is visible, ma'am.
Okay, so shall I start? Because
It's fine,
this,
ma'am.
okay. Ma'am, this title you already told to this, those who are attending. That is the plant. My title of presentation is Plant Collection and Identification for Research and Development. I selected this topic because every day I am identification, rather authentication also. Then next slide is on this about NISPER. Now NISCARE name has become NISPER, ma'am. And that is after merging the two institutes, NISTAD and NISCARE. Its name is now NISPER, National Institute of Science, Communication and Policy Research. Fine. So NISPER is a science communication for the education and research, being a premier institute of science communication and information resources. This institute published long back the Wealth of India series, which is encyclopedic dictionary of Indian raw materials, which includes monographic details of all economic plants, animals, and minerals. And Pandit Jawaharlal Nehru ji inaugurated this first volume in the 1948. Okay, I am very happy to see his 4 words on the first page of Wealth of India. Always I do show the students, those who come from schools, they are eager to see his signatures. So I really feel a high regard for them. So RHMD material, you already told that this was established in 1978 as a part of Wealth of India. Hence, we have specimens of plants, minerals, and animals. All 3 natural products samples are with us. But since we don't have any animal experts here for authentication, I don't think they require, and minerals also, just for the showcase we are maintaining them. And the acronym RHMD was allotted by International Association of Plant Taxonomy, New York Botanical Garden. Every time, because our people, even our people, those who come new, join as admin people, they ask, we are doing authentication. Is it recognized? Is it affiliated? Is it approved? So we have to show them the index, herbarium index from the New York Botanical Garden website. And we are several times proving ourselves, yes, we are not doing any activity here which is not recognized. So this is our strength, that we are recognized body. In fact, herbarium is maintained by NBRI also, and CMAP, CDRI, and even our this NCL, Pune, they are also having. But they rarely do the authentication. Several people do go to Lucknow, but they refuse it because we are busy in our work. We don't have time. You go to NISCARE. So I am attending as my duty and as my passion also. So NISCARE is publishing, madam, currently 19 research journals, and the Science Magazine, Science Reporter, Vigyan Pragati, and Science to Dunia, you know better. And this newsletters also. Next is Manoj. Ana jara kahan gaya tumne? This one. Next slide. RHMD correct botanical identity is required for research and development, because just the naming is not enough. Is common name, synonyms are also related. Because many times people bring the samples with common name only. But they have to have the botanical identity, because until unless you have botanical name, you cannot search for the literature. Go for this literature. That's why botanical name is compulsory. Even the synonyms. As you know, the plant contribute more than 60 to 70% in the development of modern medicine, directly and indirectly. Because modern medicine means that even the clinical study, they require pharmacological studies, they require pharmaceutical studies. You know, the All India Institute of Ayurveda and All India Institute of Medical Sciences, they have a Ayurveda branch. From there also we are getting samples for authentication. It has become necessary because they felt it that details are to be taken from the recognized body. Most of the plant possess several medicinal properties. However, there are substitute and adulterants. Substitute adulterants also we are getting. Madam, I am telling you, don't take it otherwise. From ITS Muradnagar, I got a sample under the name of Susurya Lapa. As soon as pharmacy head, she's pharmaceutical scientist head, and she sent the material as a Nilambo. Nymphaea nucifera. She sent me that sample. I got surprised, madam. Susurya Lapa, who has provided you? And they were, I received the petals, and she was mentioning the leaves. So this is the really, I by heart I'm sorry to say that I should not have shared this thing publicly, but it happened with me. Discovery of any active principle need to know the source, locality, habitat. To link the pattern of development active principle, you know, man, better than me that active principle development is really depends on the locality and habitat also. You may not be getting the same active principle from a high altitude if the species is found in high altitude or the species is found in the tropicals. For heart disease, mostly roots and rhizomes are used compared to leaves. Hence, correct identity through authenticated samples is essential. We get more samples on root and rhizome. I don't know, because maybe the reason, because nowadays heart diseases are increasing. So people must be working mostly on the heart diseases. That's why I'm getting the roots and rhizomes. We have very good collection here, roots and rhizome. So I'm happy to tell you that Crocus sativus has has a cure, is used to cure 5 heart diseases. Elateria cardamom, which we use daily in daily life, cures hypertension, heart attacks, even the curing the blood fat, etc. Carissa carindus, which is we used in the karanda as a pickle we use in our houses, na? So it is also useful for the congenial heart diseases. Manoj, मनोज, ये मेरा फोटो यहाँ क्यों आ रहा है? ये डिस्टर्ब कर रहा है. चलो, ma'am, actually my photograph is disturbing you or not? Hello?
No, ma'am. No, ma'am. बिल्कुल भी नहीं.
अच्छा, that's why I called in that maybe my photograph is disturbing you.
No, 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 ma'am.
Collection of plants. We are if really planting to collect fresh specimens, so a lot of preparations are required. First thing is permission from the forest department. So we have to write the forest department of the area where we are planning to visit, and collection background data. That is, we have to be prepared with the altitude of the area, possible flora types, seasons to check vegetative, flowering, fruiting stages, details of the reported species, index of common names in all languages. Because if you are going in some a place where the common name is different from the Hindi name, so we have to collect the languages, local names also. And Nisker has published a dictionary of plants, where the all common names of the all species reported in Wealth of India are available. So you can buy it very cheap book, and that will help you in collecting, knowing the various common names in various languages. So in fields, collection may be classified as per the date and place. If you are collecting, we have to classify the plants separately as per date and places, and tools which require the scissors, clippers, knife for cutting twigs, stick and hook for collection of parts from the tall trees, and field notebook. Polythene bags, old newspapers, magazines, pencil, permanent ink pen, magnifying lens, digital camera, flora book, colorful, because we have to sometimes match with the color of the flower. For say blotters, plastic polybags, and herbarium and polythene. Hot air blowers, first aid boxes. First aid box, I'll tell you the relevance of the first aid box. Topographic maps, binocular, and gum boots. Yeah, I'll share with you how the gum boots were helpful for me. And goggles, protection from the flies, mosquitoes, because in the field you will suffer from a lot from the mosquitoes, flies, and insects. 5 to 6 specimens of each species should be collected to replace damaged samples, if any, during transportation and preservation. Regular field visits are necessary to obtain information from the all stages of the growth. Safety preparations for collection. Medicine kit, I already shared with you that for nausea, fever, allergy, stomachache, diarrhea, and cuts. So first aid box is highly required. Bandage, and ointment, sugar, multi-purpose, including honeybee bites, traditional medicine, sugar, because anytime if somebody has got is suffering from diarrhea, then also sugar is required, and sugar is required because honeybee bites are there. Is the for honeybees, if they have bitten you, then sugar is required because I was bitten by a lot of honeybees in my head, because my colleague was throwing the bunch of the plants from high altitude, and I was staying in the road, and we were in the above 13 feet in the Tungnath area. So as soon as he thrown the plant on me, catch it. So I catch it, but it hit the he was stuck into a honeybees group, and they came directly to me and bitten me thoroughly on my head. So I suffered a lot of swelling and itching. So local people told me that, madam, give madam ghee and sugar. So that's why she will be how I was resting in a day in a camp, and then this sugar and the ghee cured me, and no evil was effective and nothing was effective. So salt is required because you have to protect yourself by the leeches, from the leeches, and leeches, they will enter in the gumboots. So when we removed, as soon as I removed my gumboots, I saw that a lot of leeches were in my feet. So there people said, madam, you did not put the salt in your gumboot, that's why you are suffering by leeches. So gumboots, goggles, cap, mask, binocular, they are emergency call numbers are also required for any medical aid, and wildlife update also required, because one has to be prepared that any time wildlife may attack you. Mosquito repellents really thoroughly required. Wildlife honeybees collection are the main threats for these areas. Aloe vera, as I said, that toxicity knowledge is also required, because aloe vera is toxic. How it is toxic, you must be knowing. Madam, you know? No. Okay.
Yes.
No, because aloe vera, स्किन थोड़ा स्किन को साफ करना है, थोड़ा गोरा होना है, तो एलोवेरा लेना है, तो उससे पहले you have to be careful to ooze out the only transparent the liquid which comes out of the aloe vera, because as soon as you will touch the green part of the aloe vera, it is really harmful and itching and allergic and everything. So taking this care of it, I published an article on aloe vera in Science Reporter. People and girls particularly, they must know about it, that aloe vera transparent liquid is only helpful, not the other part. So Osimum sanctum also we do take roots particularly, we should not use, because only leaves are required. I am getting roots for authentication. Maybe they are processing in other way. Otherwise, roots are mild tranquilizer and psychoactive also. Meristica fragrance. As soon as the children are born, the dadi ma will suggest, उसको डायरिया हो जाता है, तो then they will suggest, अरे जायफल और जावित्री दे दो ना, सब ठीक हो जाएगा. It's not. The quantity should be taken care of. 5 to 6 only gram may be given, not the more than that. Poisoning and giddiness and hallucination. Madam, I prepared a project on the toxicity in the plants and submitted to NMBP. And Mirabilis jalapa, which is a very common garden plant, 4 o'clock, wahi khilta hai wo, that's why that is called 4 o'clock plant. And there is one, ma'am, 9 o'clock plant also. वो 9 o'clock को ही खिलता है. So then after roasting, it looks like makhana. But it's very poisonous. Solanine, as soon as we buy some potatoes from market, brinjal and tomato, you will find that there is some green part on. some potatoes and the tomatoes also. That particular potato or tomato should not be eaten. But when I was sharing this information from with a in some meeting, the girl was from Hyderabad, and she stand up and said, "Ma'am, my dadi prefers green tomatoes only, potatoes only, and green tomatoes. She makes the chutney out of the green tomatoes. मैंने कहा the green may be the fully green, may not red part. That's why she may be using. I don't know. I'm just sharing with you whatever knowledge I gained collected from the published paper. So Delphinium denotatum jadwar, I am getting daily for identification. That is also harmful. Argyria nervosa. Argyria nervosa is seeds are hallucinogenic and the structure is like LCD. You just see. Argyra seeds are also coming to our herbarium for authentication. And Argyra, after reading the Argyra, the Environment Division of Delhi, they accepted my project that, Madam, kindly share some information with us which are having the constituent like LSD. So we have to be careful for all these things before going further studies. Limited because there is limited literature available in traditional medicine's toxicity. This is the Mirabilis jalapa, you must be knowing. And next is photograph, seeds photograph. We have in our herbarium. After puffing, they look like makhana. And this is the green wet potatoes, greenness. This should not be eaten. These are jadwar, colchicum autumnale, suranjan. And now again I have come to for the preparation of the field visits, because toxicity I told you before going to field we need to know about toxicity, and these are the instruments which we require. And this brush, the cleaning of the brush, this is the cutter, forceps, matchbox and torch. As I said, torch is very much essential. The matchbox is very much essential. Torch, the wild animals we used to get rid of, make them go away by using the torch. As soon as we put the torch in their eyes, they'll run away. And this one, the matchbox is also required, and brush. As when we collect the plant and bring in the camp, then sometimes we find a lot of it's coating of the dust or insects and something. So we need to clear them as soon as we come to our camp, because if we'll dry the leaf as it is, and file also, and then mount also, sometimes the leaves are having some fibers, or some roots are having some क्या बोलते हैं उसको, some fibers you just see that, but it's not the leaf is surface is not fibrous, but it's a coating only of the dust. So that we have to remove this from brushes as soon as we come into the camp. These are the digging material instruments for the collecting the roots. Hand lens should be there with us. And this is the field notebook. We had to have these field notebooks because these we have to collect 5 to 6 samples, and each sample should bear the same number. You got my point. These number are same for a single species. Collection of 5 samples we are having, so we have to give the same number. And this thread is to be tagged with the collection. Is it clear? And this is having the collection number. Later on, we have to fix it on the herbarium sheet. So the collection number, dates, family, genus, all these details are to be filled in the after coming in the department. So locality need to be filled up. Frequency, habitat, remarks, collector's name. Who has collected this one? So field notebook, field record in small pocket size notebook. I'll show you this notebook I already shown you. Date of collection, location, number, specimen, if possible, description of the floral parts, if possible. Flower photograph from various angles, because flowers we collect and then as soon as we come in the camp, sometimes their color is not visible as it was in the field. Dorsal and ventral view of the leaf, hairy, non-hairy, texture, leathery or succulent. Succulent leaves, I'll tell you really is required. Range, GPS data, associated species to be mentioned, at least 5 associated species to be mentioned. Status of plant either rare or common. In case found rare, then it would lead to the further studies pertaining to the protection and regeneration. You know, the rare is frequency that is rare is to be mentioned. Duplicate samples, one species that are collected on the same date and the same locality. Segregate the plants habit-wise, tree, shrub or herb. Rice preservation is the herbarium, transparent pulpet, pet and bottles. For dry preservation, herbarium is one, transparent pulpets, which I will show you, bottles and glass bottles and jars. We have preserved in these 3-4 types of material. Preservation methods, succulents is different, algae is different, bryophytes is different, bigger fruit plants and seeds are different. For succulents, apply salt if there is some milky exudate. It has to be done in the field itself, because some plants are having milky exudates and some are having transparent juice. For succulents, the succulents are to be placed in boiling water immediately as soon as we enter in our herbarium or our department. Then the succulents should be kept in the boiling water to kill the cells before placing, because otherwise the succulents start germinating in the storage places. Pyrethrum spray, organic insecticide, like it is obtained from Chrysanthemum species, that they has to be used for safety purposes. Preservation of fruit ducts and herbarium cabinets and sheets. Storage cabinets, pigeonholes. I'll show you you know better than me that its height is 45. This is 45 centimeter deep. These are the standard size of the pigeonholes, that is storage cabinets, and 31 centimeter wide and 15 centimeter high. Universally accepted, these dimensions are approved for the cabinets in vertical row. Herbarium sheets or mounting board should be 100 rags paper, because mounting boards or herbarium sheets are also standardized by recognized herbaria, and we have to follow. That should be strong and not to bend easily. I saw some of these places in university department. They just use the very common sheets for mounting. That is not enough. And this type of GSM and all these rags paper should be used, because they are sometimes moisture absorber also. But however, there had to be specimen had to be prepared before mounting. It should be 28 to 42 centimeter minus 1 plus centimeter. Genus folder should be 43 into 65 centimeter. Mercury chloride and alcohol rectified spirits we are using for before as a poisoning material before putting the plants in the jars or in the herbarium sheets. RHMD samples are preserved in a rectified spirit. Above chemicals are purchased. Madam, if at all we require the material, we are following strict guidelines, and a license is to be taken to buy this material for preservation. Making solution required experience and training. We were having a technical person especially for making this solution. I don't make this. So this is the determinate slip we are using on the herbarium sheet using. Earlier I shown you the family, number, date, names and synonyms are also to be made if we know the common Hindi name or Sanskrit name, whatever people, those who are, if they are in the south, they mention the Tamil Telugu names also. And the roots and the collector name, Dr. K. Ramamurthy, BSI Coimbatore. He collected this in 1977. Counting on herbarium sheet, a small paper envelope, which I told you, this is called fragment pockets, are attached to the sheet on the left side of the herbarium sheet on the top corner, because right side is not safe. Left side of the herbarium sheet they are used, because seeds, extra flowers and any part of the specimen are stored there. I frequently take help of this type of fragment packets, because seeds and the flowers had to be mashed thoroughly, and that's why these fragment sheets, which were maintained by our seniors here, and I am using for this authentication purpose. Poisoning precautions should be taken to protect herbarium specimens from damage by insect pests. Now, nowadays the using these chemicals is not required. So we are doing cryopreservation, means we are putting the herbarium sheets under in the cold herbarium places, that is refrigerators. We are using. Now we are putting the herbarium sheet in refrigerators, and after that we dry them and then put her in under herbarium sheet. Because so many times I do see that cigarette beetles, booklets and silverfish are very common. In herbarium cabinets, insect repellents we are putting in the naphthalene pouches. Last week only we got the cotton pouches and naphthalene balls from our admin, and we are putting them in the herbarium sheets. Molds, mildews are the common threats. This is the herbarium sheet, and this one up on the left side corner, this is the fragment packet. And the folding this fragment packet is also in needs carefully and training also, because it will never open until unless we bring out the sheet out, and then it has to be opened carefully. So, role of herbarium and museum in education. Really, nowadays our importance has increased, because authentication has become must for each and every, whether you are doing pharmacognosy, you are doing botany, you are doing chemistry. So our importance has increased now. Earlier people were not coming so frequently. Nowadays I'm not getting a time so many because ma'am, nowadays Ayurvedic, Unani and Tibetan medicines have become has become a boom. Everybody is behind the Ayurvedic medicines. No, that's why these people are coming. Even Tibia College, they never visited us. And last year they came to us. Saida madam came, and I'm sorry to say that she brought 6 students, and their botanical name was not matching at all with the Unani names. Because Unani names are different from Ayurvedic names, common names. Each and every specimen had to clear. And after that, they were in Corona, and after that, I also got the Corona. So this is the tragedy sometimes, and still last week only they brought the Nakhuna, and their botanical name was different, and it was Aegiceras. So that's why I feel sometimes okay, it's my duty I'm doing. I'm very cool. This time maybe you are listening me loudly, but I'm really cool. Correct identification for botanical name and synonyms. Until unless you give the synonyms, the old literature cannot be correlated with this one. Factors which lead to the wrong identification. Collection by unskilled person, purchase from traders. Yeah, because traders are very clever, intentionally or intentionally. They they mix the samples. That's why in my certificates you will find the line. This certificate pertains to the quality and quantity of the specimen submitted in the RHMD. Am I clear? Because I am responsible for the 50 gram only, 100 gram only. I don't bother about the kilos which you are going to study. It's your duty. Thoroughly match with the higher quantity with the lower quantity or the small quantity of the specimen you got authenticated from RHMD. That's why I regularly, correctly, don't forget to mention even our certificates are regularly same, and I just change the botanical name and all that. So factors which lead to the wrong collection and skill, purchase from third, variation or similarity in organoleptic properties of crude drugs not known to common people. Tasting is not. You know the taste of the many plants are almost same, but it is never recommended that you test it in the field itself until unless the person who is with you, local person, he will advise. हाँ हाँ आप खा के देख लीजिए तब ठीक है, अदरवाइज आप फील्ड में कभी भी टेस्ट मत करिएगा. Confusion in the vernacular names. Madam, I'm sorry to say that between indigenous system of medicine and local dialects, madam, yesterday only, I solved this problem. Common name Brahmi. I don't know क्या हो गया ब्राह्मी को. It's available in my garden itself. अरे भाई तुम मुझसे 2 ट्विग ले जाओ लेकिन ब्राह्मी को ब्राह्मी की तरह तो लाओ. क्या है कि ब्राह्मी, correct Brahmi is Bacopa. Okay, my colleague published a good paper in the Indian drugs. The correct Brahmi name, common name of Bacopa is correct Brahmi. Centella asiatica is Manduk Parni. And I am getting Meremia. Meremia is totally different. अभी कल की बात है मैडम, Meremia was submitted to me and it was Centella asiatica and she wanted to work on Bacopa. आप सोचिए, आप ये तो बहुत frequent है. और तो छोड़िए आप, मेरे पास तो in pieces में आता है ना plant. So I instead of neem I get Mahaneem, Melia directa. आपको I'll show you how I identify Melia and Ajar directa. मीठा नीम भी होता है कभी कभी, मुराया, उसमें मिक्स कर देते हैं. Substituent and adulteration of crude is common because of due to high price. So people think अरे हम इसी पे काम कर लेते हैं, क्या फर्क पड़ता है. लेकिन जब उनका पेपर सबमिट होता है ना, they get back. Mention here the authentication number. आप नेट पे डाल लीजिए मैडम, more than 200 times सुनीता गर्ग दिख जाएंगी. Because my name is mentioned in the material matter. Yeah, madam. This just see, this is the correct name, Ajar directa indica. This side picture, left side picture is Ajar directa indica, and right side is Melia Ajar directa. Can you just tell me one point? Oh, just chalo, waste of time nahi hai. So you know in neem. अजर इंटर का tilted, थोड़ा झुका हुआ है मिडरिब, और डेंटेशन इज आल्सो डिफरेंट. अभी और भी दिखाती हूँ भाई. आखिर नीम तो नीम ही है ना, थोड़ा अकड़ के रहता है ना, कड़वा है, थोड़ा अकड़ में चलता है वो, इसीलिए उसकी मिडरिब तिरछी है. और बेचारा जो महानीम है वो सीधा साधा, उसकी देखे ना मिडरिब सीधी जा रही है. और भी डिफरेंस दिखाती हूँ आपको. के फ्लावर में भी डिफरेंस है और इसकी अरेंजमेंट ऑफ द लीव्स में भी फर्क है मिलिया अजर से. ये देखिए, ये है मिलिया. अब आप सोचिए मैडम, ये आता है मेरे पास फ्रीक्वेंटली नीम की जगह, अजर की जगह मुझे मिलिया मिलता है. और ये उसकी निमोली है, नीम फ्रूट्स हैं. और I'll tell you that ये जो महानीम है बकैन, इसके फ्रूट में एक होल होता है और वो that hole can be used for making a string. उसका आप एक शो केस बना सकते हैं अपनी विंडो पे टांगने के लिए, बहुत सारे कीड़े मर जाएंगे। तो इसके अंदर इस सीड के अंदर आपको फ्रूट के अंदर होल भी मिलेगा। और ये है बेचारा अजाडियोटा इंडिका सूखा हुआ एकदम, ये है, दिस इज द नीम सीड। हाँ मैडम, आज तक एक आधा तो छोड़ दीजिए, मैं कसम नहीं खा रही हूँ, लेकिन ये आज तक मेरे पास सारका अशोका और पोलिएल्थिया लॉन्गिफोलिया की बार करेक्ट नहीं आई है, क्योंकि मुझे हमेशा पोलिएल्थिया लॉन्गिफोलिया ही आता है। और नाम क्या होगा? सारका अशोका, अशोक छाल। अरे भाई, अशोक और अशोका दोनों में अंतर है। अशोक तो सीता अशोक से आया मैडम, अशोक, इसीलिए इसको बोलते हैं सीता अशोक। और लोग देते हैं अशोका जो मास्टरी की तरह ऊपर तक जाता है। दिस इज वेरी नॉट ए वेरी फ्रीक्वेंटली अवेलेबल। सीता अशोक, सारका अशोक मेरे गार्डन में है मैडम, ये फोटो मैंने अपने यहाँ से ही ली है। और इसकी भी स्पेशलिटी बताती हूँ, एज सुन एज बार्क इज टेकन आउट ना, इट मेक्स क्विल, अपने आप ही मुड़ जाती है मैम, ये अपने आप ये क्विल बना लेता है ऐसे। और इसकी जो ये ऊपर की सरफेस है एकदम डॉटेड है। और ये आप देख लीजिए मास्टरी का, मतलब ये पॉलिएल्थिया का क्या क्या डिफरेंस है। ये चीजें नहीं सिखा सकते लोगों को, मार्केट वाले नहीं बताएंगे। उनका क्या है, उनको तो मार्केट, बहुत सारे अग्रवाल भाई बैठे हुए लोग कहेंगे अग्रवाल ही सारी बेईमानी करते हैं, ये जितने अग्रवाल और गर्ग बैठे हैं ना, ये सारे यही काम करते हैं वो पढ़ाया अग्रवाल जी, मेरे बच्चे जाते हैं तो आप उनको पोलिएल्थिया मत दिया करो मैडम। फिर अग्रवाल जी केम टू माय कैंपस, आई शोन हिम, भगवान के लिए बच्चों की थीसिस और पैसे मत खराब किया करो। पैसे तो आप जिंदगी भर बना लेंगे, बच्चों की गलती जब सुनीता गर्ग 590 एक बार लेगी, दूसरी बार भी लेगी। तो ऐसा नहीं है, ऐसा मत किया करिए। तो मैडम ये सीता अशोक कहलाता है बिकॉज सीता माता वाज सिटिंग अंडर दिस ट्री इन श्रीलंका। और आप ये देखिए मैम, मेरे पास 5 ट्री हैं और उसकी जो देखिए मेरे अभी भी गूज़बम्प हो रहे हैं, वो उसकी जो कैनोपी है ना एकदम छतरी की तरह है एकदम। तभी सीता माता उसके नीचे इतने दिन, इतने साल, 1 साल 1.5 साल बताते हैं बैठी थी, क्योंकि उसकी कैनोपी बिल्कुल उनकी छत्र छाया बनाई हुई है। वो बाप रे, मुझे क्या हो गया। तो वो क्या हुआ कि उसके नीचे ही बैठी। और एक और खासियत है, आप इस पर स्टडी शुरू करा दीजिए, क्योंकि इसका ईच एंड एवरी पार्ट starting from root to top fruit and fruit, they are useful for ladies problems. इसका कोई भी पार्ट ऐसा नहीं है जो लेडीज की प्रॉब्लम को दूर करने के लिए सक्षम नहीं है, यूजफुल नहीं है। सो यू स्टार्ट वर्किंग इट। मेरे यहाँ से लोग किलो किलो लीफ लेके जाते हैं इसकी। हमने 5 प्लांट लगाए, अपने माली को मैंने बहुत एनकरेज करा, मेरा इज्जत करते हो ना तो और प्लांट लगाओ। देखिए मैडम, अब नेक्स्ट दिखा देती, नहीं तो इसी पर अटक जाऊंगी। अभी डिजिटल हर्बेरिया व्हाई इट इज रिक्वायर्ड, बिकॉज procuring, storing, retrieval, dissemination and information data on species, specimens online, easy, quick access. Method to sharing information among users across the globe is really useful. That's why this everywhere, even the Kew Botanical Garden, BSI, they are going to this making the digital herbaria. But I don't think that that will be accessible to everybody. Local people कहाँ से जाएंगे उसको access करने के लिए? Still the physical this herbaria is to be maintained. Image and description in digital form are helpful. Quick comparison for tentative identification. It reduces risk of damage storing in several years for posterity. User can browse specimen from different countries and monitor without getting exposed to hazardous chemicals, preservative. Of course, it is required, but I don't know whether characters will be there. This is the genus folder. Color is sometimes used green, and we have used gray color. Here, left side is the species name, family name, and right side is the genus name. These are actually guidelines given to us, and this is inside the species folder. The species folder is different from the genus folder color. This is the pressing machine used on tour. When we are on tour, we used to carry this pressing machine, and inside there are blotting papers for or newspaper for drying the specimens. After tour, when we were coming to camp, then this type of pressing machine was required. So after tour, this is having screw on blotting papers or the newspapers, and we had to change next day. Fresh specimen is kept for 1 day, and next day we have to change the blotting paper or dry newspaper, and then we'll use fresh, because that that catches the moisture now. So every day we have to change the blotting paper or pressing the newspapers, and then turn the side also. Suppose we use the dorsal side, then we have to change the ventral side of the leaves. And this is our storing place. And this type of boxes we are having if the material is less for us. We are using petri dishes, and you know that we have displayed here Bitula leaf. That is Bhojpatra. So last week only we got a Bhojpatra authentication sample. And for maintaining using the space, we are using the smaller size of cabinets, and on the upside we have kept this type of glass boxes. This is the view of the herbarium, how we are storing in the cabinet box. This is the fragment, this is the naphthalene balls. We are storing naphthalene, putting naphthalene balls and putting it in the pigeonholes for protection from the silverfishes. And this is liquid preservation for the chocolate tree fruit we have displayed for attracting our newcomer, that is the student. They get attracted. Ma'am, ये chocolate tree का seed है क्या? हाँ, यही है. इसी से chocolate बनती है. So we are doing a liquid. We are putting the succulents in the liquid preservation. Yeah, this is vanilla. This is coffee beans. So now the next topic is, ma'am, time is there or no? Should I stop it here? Hello? Ma'am, should I stop it here or continue?
Ma'am, you can continue.
Okay. Because this is going out of the herbarium. This is a livelihood prospects of MSME from the medicinal and aromatic plants, or how people are using our herbarium, and for the MSME. MSME also they get authentication certificates and then go for maintaining the large scale cultivation or large scale selling. Medicinal and aromatic plants are increasingly being recognized as an important source of livelihood opportunity for rural people, and go for it. The students, those who are listening me, and don't get frightened after studying the pharmacy topic. You can go for the livelihood if you are not getting any job. Go for this providing the selling the medicinal plants samples. Large scale cultivation, you can go for selected medicinal and aromatic plants to supply in the market, university and colleges. Nursery pots, selected species are used as study materials. Area specific need B Pharma, M Pharma, MSc, PhD, because many students come here, ma'am, we can buy from your this type of this collection from your nursery. No, we are not providing, but I am opening this avenue to the students, those who are hearing me, that nursery pots selected species as study material is required many a times. orchard developments, aromatic plants for essence, medicine, insecticide and pesticides, green manure, pollution control and decoration. These are the MSME suggestions. Livelihood for the MSME, planting material, corms, cutting saplings, potted plants, genuine minute seed, that is collection from the for sales, ornamental plants, bonsai, foliage, flowering plants, season flower, establishing nursery, amenity saplings, and export and import, food, fruit and timber, indoor plants for hospitals, hotels, airports, resorts. This has been now recognized. You, as soon as you enter in a big hospital, they will show you, ma'am, we are eco-friendly, we are having indoor plants, and just see the our space. We have maintained the plants also, landscaping, these wasteland developments. And also you can go for books and books publication and green color extraction from the leaf litter, agarbatti, like CMAP is doing, and using the leaf, the dried flowers for making agarbatti and leaf litters for the extraction of green color. Rangoli colors, one Madhumata Puri lady, she is beside in nearby our office. She is collecting early morning the flowers from the temples, and she is making the rangoli colors of out of those waste flowers which are thrown by temples in the outside their temple, and she is taking an auto rickshaw and collecting these flowers and making rangoli colors and agarbatti. So, and lotus cultivation in fields, madam, this about the lotus cultivation has also been studied by the our HBT lab, and they have tried the lotus cultivation in the fields. It's not limited to the only ponds. Tries to crush flowers from temples, hotels across the environment department funding. Environment department, she got the funds from in the initial stage from the Department of Environment for collecting trash, for maintaining trash to cash. Vermicompost, organic manures, leaf litter, vegetable waste. Vegetables waste is used by IRI, and collection of non-wood forest products, waste, fruits and leaves. Active principle are also being accepted from the leaf waste. Biomass, fuel and briquettes, mushroom cultivations. Floriculture, sericulture, textile industry, corn, this one. Natural dyes, progression fodder, organic matter, disinfectants, herbal, neem-based and all. Natural shampoo, tooth powder, henna powder, dried vegetables, food grain preservatives. Training is being given by our CFTRI branch, CFTRI Hyderabad and Pantnagar, Ahmednagar, Nagpur. They are all university, agriculture university. They are also providing the training on this such type of products. Flower powder, flower power, edible medicinal flowers. As I initially I told you that I'm not getting much of the flowers for authentication, but if we will tell them, we share them, we make them aware of that the flowers are having some medicinal properties also, and that's why they are edible. From my eyes, I saw my neighbor while purchasing some flowers, which I got surprised that why she's purchasing these flowers. Edible flowers in kitchen. Okra, lady's finger flowers are eaten in soup in Africa as a source of calcium, iron and vitamins. So, madam, are we using this okra and lady's finger flowers in our kitchen as soup? Tell me yes or no.
मैम भिंडी फ्लावर होती है उसका ये नहीं
हाँ
पता।
वही भिंडी के फ्लावर तो
हाँ अच्छा मैम फ्लावर
करते हैं आप उसके यूज करते हैं फ्लावर?
नहीं मैम फ्लावर तो नहीं यूज करते हम।
शेयर करा, दैट्स व्हाई आई एम शेयरिंग कि ओकरा मतलब भिंडी फ्लावर्स, लेडीज फिंगर जिसको कहते हैं, दे आर ईटन इन अफ्रीका एज ए सोर्स ऑफ कैल्शियम. दे आर मेकिंग सूप आउट ऑफ इट. ट्राई तो करके एक स्टडी टॉपिक दे दीजिए ना किसी बच्चे को,
मैम इस बार देंगे हम आपके साथ ज्वाइन कराएंगे स्टूडेंट को हमारे साथ मैम कोऑपरेट करते रहिए आप।
हाँ, देखिए एबुटेलॉन इंडिकम बहुत ही कॉमन वो है, ये वीड हमारे यहाँ भी कैंपस में ही होगा, बल्कि मैंने अपने माली को लगवा दिया था। उसको कंघी बोलते हैं, झंपी भी बोलते हैं, उसके फ्लावर्स एडिबल हैं। और बॉम्बेक्स सीबा सिल्क का तो पता ही होगा आपको, पक्का पता होगा। सिल्क कॉटन क्या हुआ? हाँ, सिल्क कॉटन ट्री जानते हैं ना आप तो? सैंपल
हाँ जी
सैंपल तो मैडम जरूर खाया होगा। Flower buds and fleshy calyx are used as vegetable. They are astringent and cooling, also applied on the skin troubles. They provide exciting color and flavor to everyday meals. Its flavor are vary from cucumber like peppery. मतलब उसका जो कुकुम्बर से उसका टेस्ट मिलता है। Then Bombec Sibaka, that's why they can be used. Edible flowers contain antioxidants, minerals and anti-inflammatory activity. मैडम आप बता दीजिएगा जब मुझे बंद करना होगा।
मैम टाइम वैसे हो गया है मैम।
अब मैं और कर दूं, खत्म कर दूं?
हाँ जी मैम, हाँ जी
ठीक है, okay. Then thank you very much for listening me, and these are the some of the fruits I'll show you. This is our campus. Fine, okay. Okay, thank you. Sorry for taking your time. इतना ज़्यादा लिया।
Thank you so much, ma'am.
Thank you so much, ma'am, for gracing our FDP with your presence. Your talk was simple yet very informative, and it was full of relatable examples and practical aspect we somehow, as a researchers, are not aware of or we neglect sometimes. The house is open for discussion. If anybody has any query or any doubt, they can ask ma'am directly.
Good morning, ma'am. I Dr. Sonia here. Ma'am, actually, I want to ask: is there any site or any Google we can Google before sending a sample for the authentication, so that we can match with that? Because I have remember when I'm doing PhD on Acacia auriculatum. Then I sent sample to you, and you message me that Sonia, इसमें कटोरी नहीं बन रही. This is not Acacia auriculatum. Then still when I'm moving on the roads in bus, I have always seen इसमें कटोरी बनती है, कर लो. मतलब मैम मुझे आज भी
I'll show it again. Because Acacia auriculatum you are telling ना?
Ma'am, I sent sample 3 to 4 times because but मुझे लगता है वो पंजाब में होता नहीं है शायद. Sample authenticate नहीं हो पाया था.
Okay, message me. I'll show you the photograph from herbarium, and I'll try to share with you the distribution.
Then उस टाइम में बहुत टेंशन में थी, पर अब मेरी PhD हो गई है.
अच्छा, तो फिर आप टेंशन की जरूरत ही नहीं है, शेयर भी करने की जरूरत नहीं है. Then I'll show you.
Okay, ma'am.
You know that if this type of problems come, they should ask me that it is where it is available. So distribution is available on the seats also. So I'll share with you. Okay, then next question. Is any query?
Ma'am, there is no query in the chat box too. I think the lecture was very clear, very. Dr. Manpreet wants to say something.
Okay. Welcome.
Manpreet ma'am, you can unmute yourself. Ma'am, the question is how flowers of okra can be consumed?
How the
flowers
okra?
of yes.
Okay, I'll tell you because I have to open my slides then.
Hello, ma'am.
Hello.
Ma'am, how we can consume okra flowers as a source of calcium?
So I'll tell you how it is. It could be consumed by as a vegetable or mostly as a vegetable may be used.
नहीं वेजिटेबल तो मैम कैसे बनाएंगे उसको, कैसे उसके फ्लावर्स को?
भाई फूल को काट के और तेल में और नमक में छौंक देना.
ठीक है मैम.
Otherwise, I'll जहाँ से मैंने information ली है, I'll tell you the full paper. I'm not telling from my side. I just collected from the research papers.
Okay.
Okay, thank you very much, ma'am. It was very nice and wonderful session. Now it's time to felicitate Ma'am Sunita. Kindly accept our token of love, ma'am.
I read it. Okay.
Thank you, ma'am.
Thank you. You are most welcome to visit our herbarium. And anytime you have put me questions, I'll try to solve them.
Okay, ma'am. Next, we will visit this care CSI very soon with our students. So, ma'am, thank you so much for your encouraging talk. And this encourages us to get take a collection of more and more plants. Even I also want to attend you for the authentication. Please accept me.
No, for that particular plant which you raised the query, you just send me the picture of it, and then let me see what was the problem. I'll recollect it. I don't recollect several times that who sent me and what was the wrong thing and all that. So you share with me. I'll go. I'll see. But at that time, refusing the wrong sample or right sample, I do study a lot. It's not unnecessarily I say that this is wrong.
Okay, ma'am. Thank you so much, ma'am.
Welcome.
Thank you, ma'am.
Do visit to our museum, and you will see from your eyes. I just leave the student go in the herbarium and get identified from yourself.
Okay, ma'am. Thank you so much.
Thank you, ma'am. So our second speaker has already joined, and I feel privileged to invite the second speaker of the day, Dr. Vinod Tiwari. Dr. Vinod Tiwari is currently working as Assistant Professor of Pharmacology in Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, Varanasi. Dr. Tiwari has completed his B.Pharm from HNB Garhwal University, Srinagar, Uttarakhand in the year 2004, followed by M.Pharm in Pharmacology from NDMVP College of Pharmacy, Nasik, University of Pune in the year 2007. After completing his M.Pharm, Dr. Tiwari has worked in Nicholas Piramal Research Center, Mumbai, where he contributed immensely in the drug development project related to pain, inflammation, and arthritis. He has been my senior at University Institutes of Pharmaceutical Sciences, Punjab University, Chandigarh. He joined UIPS in 2008 for pursuing his doctoral studies, and after completing his PhD in the year 2012, he moved to United States to carry out postdoctoral research at the Johns Hopkins University Schools of Medicine, USA. Before joining IIT, Dr. Tiwari has served as Assistant Professor of Pharmacology and Toxicology at NIPER, Ahmedabad. Dr. Tiwari is working in the field of pharmacology and neuroscience from the past more than 15 years. His research interest is focused on behavioral and molecular neuroscience dedicated to understanding various neurodegenerative disorders and the signaling pathways involved so that better and safe drug candidates can be developed by targeting the disrupted neuromachineries. His postdoctoral research work with Professor Srinivasa N. Raja, Director of Pain Research at Johns Hopkins University, USA, was focused on dissecting the key neuronal circuits involved in pain relief and drug abuse, so that better analgesic drugs, which are devoid of drug addiction, can be developed. Dr. Tiwari also explored the role of mu-delta heterodimers in peripheral opioid analgesia and tolerance. Dr. Tiwari has contributed immensely in the field of neuroscience with more than 56 research publications, 26 review articles, and 4 book chapters in peer-reviewed high-impact journals. His research work is rapidly being cited all over the world with more than 3,600 citations, and his H-index is 33. The CV is very long. I will cut it short because sir has limited time. He has requested me earlier to. So it has been an honor to have you, sir, today, though virtually. We wanted to have you in person here, but it has been a privilege for us to hear from you, sir. He will be going to speak on pain, a blessing or curse, a scientific perspective. Over to you, sir.
Thank you. Thank you so much, Parmeet. So a very good morning to all the dignitaries, faculty members, and dear students. First of all, I would like to convey my heartfelt thanks to Dr. Parmeet for the kind introduction and inviting me to deliver the talk for this faculty development program going at Khalsa College of Pharmacy. So it is my pleasure to be here today and interact with you all. I request, the attendees today, feel free to, interrupt me in between if you have any queries, any doubts, and feel free to, interrupt and ask the questions. Yeah. So can I share my screen, please? Okay. So I thought it's a faculty development program, so I will talk like very general about pain, because that's my field of research. And the another issue is that this is something which is very, very common in our society all over the world, but most of the time we keep on ignoring the pain until it becomes chronic in nature. So today I thought I will try to sensitize the audience about this one of the most, I would say, the disorder which we have ignored but is very much prominent in our society. So we'll try to understand whether it is a blessing or a curse, and what are the mechanisms which plays a role in pain, and what are the different screening models which are available with us for the screening of different drugs for the treatment of pain. So before starting my talk, I would like to pay tribute to Mahamana Pandit Madan Mohan Malviya, who has founded this beautiful temple of learning, Banaras Hindu University, also known as the seat of learning, where we are sitting today. And those who don't know, Banaras Hindu University is the first institute, not only in world but in whole Southeast Asia, who has started the pharmacy education. So we are the first, not only in India but the Southeast Asia, where the pharmacy education has been started. So this is the recently we completed the 100 years of existence, and this is the postage stamp which was released by the government of India on completion of the 100 years of BHU and IIT BHU. So yeah, so when we're talking about the pain, some of the most common queries are, what is pain? This is something which we experience in our daily life, but sometimes it is difficult to explain, like whether it is good or whether it is bad. So anybody from the audience would like to say something about it, whether it is good or bad? Okay, so we'll talk about it in the coming slides. So yeah, pain is whether it is physiological or pathological. So after completing this talk today, we'll be able to understand about the different perspectives of the pain. How do we feel pain? How to assess the pain in animals? Because animals are they are non-vocal. We don't understand their language. So most of the drugs which are currently in clinic or which are currently in the process of drug development, they go through this the preclinical development phase. So whether it is good or bad, right? So before I jump into this like notion, this is a quote by Swami Sivanand. Oh man, you curse me, blame me, you hate me and frown at me. You think I'm cruel and heartless. You try to slay me with anesthetics, with chloroform and bromides. You attack me with sedatives and opiates. You phone the doctors, you run to the hospitals, you fly to Vienna and hill stations, you wire to your friends and relatives, you approach the sands of the Himalayas for remedies and herbs, you do Mahamrityunjay Japa and Havan, you burn incense and pray to kill the teacher who warns you, who comes to help and bless you. I'm not your enemy, I'm your friend. I'm your guide and a silent teacher. I'm the pain, the best thing in this world. So most of the time, we try to blame, the pain, this is, nobody like to have pain, right? So if we talk about, give me 1 second, I want to move this here on the side. Okay, fine. Okay, so International Association for the Study of Pain, the head office is in Washington, DC, United States. So according to ISP, pain is an unpleasant sensory and emotional experience which is associated with actual or potential damage. Why we are calling it as sensory and emotional? Because we feel pain whenever we are getting any sensory stimuli, like when a sharp object touches us, right? Whenever we have any, whenever we touch a hot cup of tea, we feel pain, right? So those are the sensory component, right? But when a person, feels pain, for a prolonged period of time, and the pain becomes chronic in nature, right? It leads to certain maladaptations in the central nervous system of the patient, which leads to the changes in the reward circuitry and other emotional aspects of the patient. So that's why, pain has two components. One is the sensory component, other is the emotional component. 1 second. So whether pain is good or bad, I would say it is both. As we discussed in the last quote, right? The pain is, is like a teacher who tells us, right? For example, if you're taking a hot shower of water, okay? And if you cannot feel how hot is the water, what will happen? You will end up, burning your skin because you cannot feel how hot is the water, right? So acute pain is always good and you can say protective in nature. It act as an alarm, you can say, that ultimately has a protective role, which induces the avoidance behavior in us and limit the potential damaging consequences. If we are, approaching any sharp object, then our senses, tells us, the pain tells us, okay, this is something which is, potentially damaging to our body. So we will take the proactive step to avoid any damage because of the pain, right? And if we can't feel it, then ultimately we will damage, our tissues. So the drugs which are, used for the treatment of chronic pain, actually, we call them analgesic, right? Like paracetamol is used for, fever and pain, ibuprofen is there. These are the non-steroidal anti-inflammatory drugs which are available in the clinic, right? There are certain strong analgesics are there, like morphine drugs are there, then gabapentin is there, which are available for the treatment of chronic pain condition, okay? So these are the agents which selectively release the pain by acting in the central nervous system or by modulating the peripheral pain mechanisms without significantly altering the consciousness of the patient. So what does it mean without significantly altering the consciousness of the patient? Why it is important? Because the drugs, which are in the clinic right now, for example, I would say morphine, right? One of the side effects of the morphine is sedation, right? The patient, after taking the morphine like drug, they cannot, drive. It is always recommended by the doctor, physician, you don't drive. You don't work on heavy machinery after taking this opioid drug because they alter your consciousness. And the drugs, for example, anesthetics, right? These drugs, they put us on sleep. So whenever we go for any minor surgery, we go for local anesthetics, right? When we go for some major surgery, we go for the general anesthetic administration, right? So these anesthetics, they alter our consciousness. Definitely, we don't feel pain when we undergo any kind of operating procedure in our body, right? But at the same time, we are also losing the consciousness. So the condition for the good analgesic or the painkiller is that they should be able to suppress the pain, but does not alter our consciousness. The person should be awake, should be able to function in day-to-day life. Okay. Now the next question is, how do we sense pain? So whenever we encounter any tissue injury, If you see this, picture, 1 second, let me my laser. Okay, if you see this, diagram, it's a very simple diagram, not to go into like two complexities. Whenever we encounter any damage in the peripheral nervous system, our tissues have, nociceptors, just like we have the receptors by which we can taste the food, right? Or we can smell any kind of, like stimuli, right? In the similar manner, our peripheral, tissues have the nociceptors, which can actually sense the, or, different kind of stimuli, pain stimuli, you can say. So whenever we encounter any tissue injury, these nociceptors, they get activated, which are present in the peripheral nervous system, and they transfer this, pain information. We call it pain information because it's a kind of information which is transferred from the peripheral nervous system to the spinal cord. But on the way, there is DRG neurons, dorsal ganglionic neurons, which act as a primary sensory neurons. And from DRG neuron, this information get transferred to the spinal cord, which act as a relay center, where this pain information gets modulated. Means the actual pain information which supraspinal brain regions will receive maybe more or less. So there is a kind of mathematical calculation which occurs in the spinal cord. We'll come to that discussion in the later part of the slides. Okay. So the ultimate output, or the ultimate, the total pain information which goes to the brain from the spinal cord is different from what we perceive from the peripheral nervous system to the spinal cord. And when this pain information goes to the brain, as we all know that it is the CPU, central processing unit. This will tell our body what proactive step has to be taken to avoid that damage. If the damage is really credible, our brain will give them, the command to immediately, withdraw from that, stimuli, just to avoid the tissue damage. So this is how we perceive the pain, and we take the proactive step to avoid it. Now, pain receptors, as we discussed, these are the type of receptors which are present at the end of sensory neurons axon and respond to the damaging or potentially damaging stimuli by sending possible threat signals to the spinal cord and to the brain. And if the brain thinks the threat is credible enough, it will create a sensation of pain to direct the attention of the body to that particular body part so that the threat can be mediated. This whole process is known as nociception. In simple terms, you can call it pain, but in scientific terms, we call it nociception. That's why the drugs which suppresses the pain are known as anti-nociceptive drug, or you can call them analgesics. Now, in this slide, what I'm trying to tell you, like how actually the pain information is perceived or transferred from peripheral nervous system to the central nervous system. So what do you see here in this picture? These are the peripheral nerve terminus. This is the peripheral nervous system, on the top, and in between is the sensory ganglion, and then this is the central nervous system, which consists of our spinal cord and the brain. So the nociceptors, they are present on the free nerve endings, or the nerve endings may be encapsulated, or some of the sensory cells which are present, in the tissues, in the skin terminals, or the peripheral processes. So all these, nerve endings and the peripheral process are having the nociceptors, which are responsible for sensing the pain, you can say. Okay. And then this pain information gets transferred from these, receptor to the DRG neurons, sensory ganglionic neurons. Okay. And from the DRG neuron, that information further goes to the spinal cord and the brain. So the peripheral terminal of the mature nociceptor is where the noxious stimuli are detected, and noxious means painful, and they transduce it into electrical energy. And when the electrical energy reaches a threshold value, an action potential is induced and driven towards the central nervous system. Just like we have our own language of communication in Hindi, Punjabi, or English, right? The neurons and the cells, they have their own mode of communication, and they communicate in terms of electrical signals, action potential, right? So when this action potential reaches a certain threshold, it drives the towards the central nervous system, and this leads to the train of events that allows for the conscious awareness of the pain. So this is where we actually start feeling the pain. And can you imagine all these processes are happening within fractions of milliseconds? Otherwise, we cannot protect ourselves, right? So nature is so wonderful, all these complicated, signaling pathways are being activated and the information is being conveyed to the brain, and the brain is taking all the proactive steps to prevent us from the damage. All these things are happening within fractions of milliseconds. So now the next, is what are the different kind of nociceptors which are present in our body. So when we touch anything which is hot, we can feel it is hot, right? Whenever any sharp object touches us, we can feel that kind of stimuli, whether it is some mechanical or thermal or chemical. So based on the kind of stimuli, we have different kinds of nociceptors which are present in our peripheral nerve terminals. So for the thermal pain behavior, when we hold a cup of tea or a coffee, or when we take a shower from with the hot water, right? We can actually feel how hot is the water or how hot is the cup of tea. So how do we feel it actually? This is because of the presence of transient receptor potential vanillate 1 nociceptor, which we also known as TRPV1 nociceptor. If you just go to the 2021 Nobel Prize in Physiology or Medicine, that has been given to the Professor David Julius in the University of California, and that prize was given to David Julius for the discovery of this nociceptor TRPV1. Okay, and the cold sensation, you might have felt that cold sensation when you take the menthol in your mouth, right? So that cold sensation is because of the activation of TRPM8 channels or nociceptor present in your mouth and tongue. So this is how we feel hot and cold sensation. In the similar manner, we have the nociceptors, which can sense the changes in pressure or any mechanical deformation or the incision which can break our skin surface. So these receptors are known as mechanical nociceptors, and the example is TRPA1. This is transient receptor potential anchoring 1 receptor, which detects the mechanical nociception. Then there's another category of nociceptor which we also call as chemical nociceptors, because they can react to the changes in the sensation because of any chemical such as capsaicin, right? Which we all take capsaicin in our diet, right? So what does capsaicin does? Capsaicin can activate the TRPV1 nociceptor. So that's why if you know that when you eat excessive capsaicin, what happens? You have kind of burning sensation in your mouth, and that burning sensation is because of the activation of TRPV1 nociceptor in your mouth. Okay, and another example is spider toxin. Spider toxin also activates the TRPV1 nociceptor. Now there's another category of nociceptor which we call as sleeping or the silent nociceptor. Why we are calling it as silent nociceptor? Because they do not react immediately to any kind of potential noxious stimuli. They do react once the damage has actually occurred, because it has some different role to play in our body. Okay, so they do not respond at all to thermal, mechanical stimuli unless injury has occurred. These are typically referred as silent or sleeping nociceptor, because their role comes only at the onset of inflammation to the surrounding tissue. Okay, now there's another category of nociceptor which we call as polymodal nociceptor. As the name indicates, polymodal means these are the nociceptors which can detect more than one stimuli at a time. For example, TRPV1 nociceptor, they can detect the thermal stimuli, they can detect the chemical stimuli. So they can perform more than one function, or can detect more than one stimuli at a time. So that's why we call them polymodal nociceptors. Okay. Now what I'm trying to show you in this slide is a gentleman, and his name is, Professor Michael Katrina. He's a professor of, neurosurgery and biological chemistry and neuroscience at Johns Hopkins University. He's a sensory neurobiologist with a focus on molecular basis of pain and temperature sensation. So why I'm sharing, his details with you, because when I was working in Johns Hopkins, I had an opportunity to work with, Mike, and actually, the Nobel Prize which was awarded last year in 2021 to David Julius was the PhD work of the Michael Katrina. So he was the first student of David Julius who has started working on the TRPV1 nociceptor. After Mike, many other students, continued that research on TRPV1 nociceptor in the lab of David Julius, and finally, last year he got the Nobel Prize for his, this work. So this is one publication, like our collaborative work. When I was in Hopkins, we were collaborating on certain projects where we studied the keratinocyte stimulation, whether it is enough to evoke the nociception or pain in animals, in mice or not. So we performed some condition-based preference studies for them, and this was our collaborative work with Michael Katrina. Okay, so yeah. So now, you might have an idea about what is physiological pain, what is pathological pain. Physiological pain is always acute in nature and very much required for our body, for our protection, right? But when the pain becomes chronic in nature, for example, in the case of certain injuries like, some accidents or spinal cord injury or diabetic neuropathy or arthritic pain and migraine, all these are, disease condition where the pain becomes chronic in nature and unbearable by the patient, so that he has to undergo the treatment by the physician. So those kind of conditions are known as the pathological pain. And this is the situation where we need the medical intervention. So here if you see in this slide, you will see these two gentlemen are here, So one is Melzek, another one is Wall. So the reason for discussing these two scientists here, because these are the two scientists who has revolutionized the pain and the sensory biology research by their discovery or by their theory of gate control theory of pain. So this theory of pain asserts that the non-painful input closes the gate to the painful input, which prevents the pain sensation from traveling to the central nervous system. So therefore, stimulation by any non-noxious input is able to suppress the pain. So this theory was proposed by Melzek and Wall in the year 1965, and they have published their theory in the Science Journal, which is one of the topmost journal in science, and they got the Nobel Prize, for this research actually. And this theory is considered as one of the most influential theories of pain because it provided the neural basis for pain perception and modulation. So according to this theory, actually, both Melzek and Wall, they propose that there are two kinds of fibers which are present in our, body. One are the thin fibers, which we all call as pain fibers, and other fibers are large diameter fibers, which are responsible for sensing the touch kind of sensation or when there are any changes of pressures or vibration. So these kind of, sensation we can distinguish actually, and this is possible because of these two kind of fibers which are present in our body. And they are responsible for carrying this information from the site of injury to the two destinations in the dorsal horn of the spinal cord. So these are transmission cells which are present in the spinal cord, and the function of the transmission cell is to transfer this pain signal to the brain from the spinal cord. And there's another kind of interneurons which called as inhibitory interneurons, which are also present in the spinal cord, and their function is to impede the transmission cell activity. So there is a balance between the transmission cell and the inhibitory interneuron, which decides that, how much pain information will be transferring to the higher regions in the brain. And that will decide whether we will feel the pain or not. So the thin fiber activity, it always impedes the inhibitory cells and will allow the, transmission cell to fire more, and we will feel more pain. While on the other hand, the large diameter fibers, their function is to inhibit the transmission cell activity, which means when the large diameter fibers will be activated, the transmission cell activity will go down and we will feel the less pain. So that is the reason why more large fiber activity means less pain is felt. Okay. So based on this, we have classified these fibers into two categories: nociceptive fibers and the non-nociceptive fibers. So the fast and relatively thick and myelinated fibers are the A delta fiber, and their function is to carry the messages, the information very quickly, with intense pain. While on the other hand, there are another kind of fiber which are present, which are unmyelinated in nature, which means they don't have the myelin sheath on them. That is the reason why they are slow conducting in nature, because myelin sheath helps in the transmission of the information, in this case pain information. So that is the reason why C fiber are slow conducting in nature, but their function is to carry the long-term throbbing and chronic pain. While on the other hand, there is another category of fibers in our body, which are large diameter A beta fiber, and we call them non-nociceptive fiber because they do not transmit the pain stimuli and inhibit the activity of firing by the A delta and the C fiber. So it is a journal, I would say very obvious when we feel any kind of pain, what is our first response? Or whenever we touch something which is very hot or which is very painful, what is our journal, like first reaction? We generally shake our hand like this or we rub our hand. So why we do like that? Because this rubbing and shaking of hand, it activates the large diameter A beta fiber, and once the A beta fibers are activated, they will suppress the activity of the A delta and the C fiber and will inhibit the pain. So this is the circuit diagram, just to tell you how this gate control theory of pain actually works. So these are the inhibitory neuron. This is just a kind of circuit diagram, or you can say virtual diagram, which is actually exist in the spinal cord. So both the inhibitory interneuron and the projection neuron, they are present in the spinal cord. In this case, you see the gate is open here. We are talking about one virtual gate which is present in the spinal cord. Okay, and these are the A beta fiber, okay, which are also supplying to the spinal cord, and the C fiber, which also supplies to the spinal cord. And depending upon which kind of fiber is activated, it will decide whether the gate for the transmission of pain information from the peripheral nervous system to the central nervous system. So in this case, if you see here, the A beta fiber are blocked. There is a cross, red cross, right? But the C fiber are activated. So we know from the previous discussion, the C fiber are nociceptive fiber, which are responsible for the throbbing kind of pain. But A beta fiber are non-nociceptive fiber, which inhibit the transmission cells, right? So when the non-nociceptive fiber are blocked, what will happen? Whatever pain information is coming through the C fiber, it will go to the projection neuron. It will go to the transmission cells. These are the transmission cells. And from here, this pain information will go to the higher centers in the brain and will feel the pain, right? But on the other hand, when the this A beta fiber is activated, which is non-nociceptive fiber, what will happen? It will closes the gate for the nociceptive fiber, and whatever pain information is coming from C fiber or the A delta fiber, this transmission cell, will be inhibited, and the gate, this gate, which is carrying the pain information from the peripheral nervous system to the central nervous system, will be closed. So either we will feel less pain or no pain, depending upon the input from the peripheral nervous system. So this is how this virtual gate control, how this gate control theory of pain works, which is given by the Melzack and Wall in 1965. And these inhibitory interneurons, they consist of GABA receptors. We all know that GABA receptors are inhibitory in nature, and in transmission cell we have glutamate receptors, which are excitatory in nature. Okay. Now, when a patient who is suffering from chronic pain, when the patient goes to the doctor to discuss about his problems, so he always suffers from two kind of component, as I mentioned. One is a sensory component and the other is the affective component. Most of the time, physicians, they are able to take care of the sensory component, but the affective component of pain is mostly ignored and unattended. So the symptoms which patient generally complains of, during the discussion, is the allodynia and the hyperalgesia. These are the two most common symptoms of the clinical pain, I would say. So do you feel that wearing a shirt or wearing the socks is painful in nature? No, not at all, because shirt and socks, these are non-painful stimuli, right? But in case of the patients who are suffering from chronic diabetic neuropathy, for them, even wearing a shirt is highly painful. So when non-noxious stimuli become noxious in nature, or you can say when non-painful stimuli becomes painful in nature, we call this condition as allodynia. But the situation where less noxious stimuli becomes more noxious in nature, we call it hyperalgesia. And if this is because of mechanical stimuli, we call it mechanical allodynia or mechanical hyperalgesia. And if it is because of the thermal stimuli, we call it thermal hyperalgesia. So may I know how much time do I have so that I can, decide how much to curtail?
So
Do we have another lecture?
You can continue at 2:20, sir.
Okay, thank you. So when the clinician, they, see the doctor, doctor generally, they ask, they provide certain kind of stimuli in the form of worm-free hairs or another kind of stimuli, and they ask the patient, how much, pain do you feel when we are touching you at the, particular point. So based on the response from the patient, doctor scores and try to understand the intensity of the pain with which patient is going through, so that he or she can decide, which pain medication to start. Okay. So if there is no hurt after giving the stimuli, doctor or the clinician generally gives a 0 score. If it hurts a little bit, it goes for either a 1 or a 2 scoring. But if it hurts a little bit more, the scoring goes to 2 and 4. And similarly, when the patient complains of, hurting a lot, or the pain is too much, like even if with just a finger touch, patient says that, no, this is something which I cannot bear, the scoring goes up to 8 or 10. So this helps doctor to decide the intensity of the pain and to figure out what kind of drugs to be given to that particular patient. Okay. So, this is the classification of pain based on its origin, based on the pathophysiology, So pain can be nociceptive in nature, or it can be neuropathic in nature. We know that nociceptive pain is because the damage in the tissues, and when these tissues, they start sending pain signals to the central nervous system. But whenever there is any damage to any nerve, like whether it is sciatic nerve or the spinal nerves which are present, in the near the spinal cord, the pain we suffer through is neuropathic in nature. And both kind of pain are distinct in terms of their pathology. That's why they both require different kind of treatment approach. Okay. And again, depending upon the tissue from which pain is perceived, it can be somatic in nature or visceral in nature. Again, if it is from the peripheral nerves, we call it peripheral neuropathic pain. If it is central in origin, like from spinal cord or because of damage in the brain regions, we call it central neuropathic pain. For example, stroke pain. Now, this is just to brief about the drugs which are currently available in the clinic for the treatment of different pain disorders. So we all know that analgesics are the drugs which act by various ways, either in the peripheral nervous system or in the central nervous system. So opioid drugs are there, right? They bind to the mu, delta, and the kappa receptors in the brain, in the spinal cord, and even in the peripheral tissues, because these receptors are present ubiquitously, in all the body parts. And once they are activated, they start the signaling, which leads to the pain relief. The non-steroidal anti-inflammatory drugs are there, which helps in the treatment of, you can see some kind of, acute pain kind of situation, like COX inhibitors are there, COX-1 and COX-2 inhibitors, because they work by inhibiting the prostaglandin and thromboxane synthesis. Now, this was all about the physiology and the pathophysiology, which is associated with the chronic pain. I thought, it would be useful for the audience if we, just discuss a little bit about the different screening models which are available in the, labs for the assessment of drugs for the treatment of pain. So all these, tests we are using very routinely in our lab. And if some of the students, need any help in the screening of analgesics or drugs for the treatment of any kind of pain, they can get in touch with us. But I have not, included all the models just because of the lack of time, but just few models just to brief the audience, about the drug discovery techniques. So these are the different models of inducing the neuropathic pain in animals. So they are just the replicas of what patient encounter when they have the sciatic nerve injury, when they have disc sleep kind of situation where the doctor says that you have to undergo a surgery to remove your disc which has slipped and compressing the spinal cord or the spinal nerves, which is leading to the radiating kind of pain from the all the way from the back, sometimes to the legs, and sometimes all the way from back up to the neck. So that kind of situation is, is neuropathic, So for that, we have different models in our lab. And depending upon the aim and objective, we can use any of these models to model the neuropathic pain in animals. So the first model is the exotomy model, where we go for the complete sciatic nerve transaction. Okay. And this can be done in rats. Now, the another model and the widely used model is the chronic constitution injury model, which we are using in our lab as well. So in this model, if you understand the anatomy and physiology of the spinal cord and the spinal nerves, it will be very easy for you to understand, how these models are done in animals. So this is, what you see here is the vertebra and the spinal cord just beneath the vertebra. Okay. And these are the different spinal nerves coming out from the lumbar region of the spinal cord, like L3 spinal nerve, L4, L5, L6. And this swell part which you are looking here is the DRG neurons. In short, we call them DRG neurons. These are the primary sensory neurons. So we call them dorsal root ganglia. Okay. So when these nerves, they come out of the spinal cord, they merge together and lead to the formation of sciatic nerve. This sciatic nerve, the function of the sciatic nerve is to supply the hind limbs. In our case, legs. In case of animals, it is hind limbs. Okay. And then further, these nerves, get trifurcated into sural nerve, tibial nerve, or the peroneal nerve. Now, depending upon the kind of nerve you are ligating, you can model different kind of neuropathic pain in animals. So in this case, in case of chronic constriction injury, we put 4 loose ligatures around the sciatic nerve. This model can be done in both rats and mice. Here you see sciatic nerve is the merger of L3, L4, L5, and L6 nerves here. This thick one is the sciatic nerve. Okay, so these 4 dots which are looking at in this picture, these 4 dots are actually the 4 ligatures. Okay, and what do you think, why we are ligating, and what will happen if we ligate too tight or too loose? If you ligate too tight, what will happen? Because our purpose is to create a model of neuropathic pain. So we want to restrict the blood supply through the sciatic nerve. There's a small blood vessel going underneath the sciatic nerve. So when you put the loose ligatures, you are restricting the blood flow in the sciatic nerve. But if by mistake, if you ligate it too tight, what will happen? You are going to block the complete blood flow, and all the tissues will die in the absence of blood supply. In that case, the animal will be paralyzed. So instead of creating the model of neuropathic pain, you will have the model of paralysis. That is not what you want, right? You want animal with hypersensitivity, means with pain, so that you can test them for different drugs. Okay. And if the sutures are very loose, in that case, the blood supply will be normal as before. So animals won't feel any kind of pain. So you put at least 3 to 4 loose ligatures around the sciatic nerve to restrict the blood supply and model the pain in animals. This is another model which we call as partial sciatic nerve ligation. This is also known as the Selger model. So in this case, we tightly ligate 1/3 to half of the sciatic nerve and restrict the blood supply again. This can also be done in rat and mice. There are other models of neuropathic pain, and it's spinal nerve ligation, where we ligate the spinal nerves, individual spinal nerves, instead of ligating the sciatic nerve. So we can ligate the L5 nerve or the L6 nerve, right? And then we cut it. Why we cut it after ligating? If you already ligated, what is the need to cut it? Because if we don't cut it, what will happen now? Sometimes they have regeneration, they sprout out, right? So if you don't ligate it after cutting, then they will make the new connection, and the pain will not be long lasting. When you want to model the pain in animals, you want to have a model in which animals have a chronic pain, at least for 1 to 2 months. Okay, so that you can study the pain pathophysiology and the effects of the drugs. But if by mistake you cut the L4 nerve, the animals will be paralyzed. So this kind of models, they need a lot of expertise, sometimes the need of surgical microscope as well. Okay. Now, after, inducing the pain in animals, you can, how to measure the pain in animals? That is the next question. Because animals cannot, tell you how much pain do I feel, right? Just like patient tell to the doctors? In that case, we have different kind of, apparatus or techniques by which we can measure the pain in animals. Hot plate is one of them, tail flick, pain or withdrawal latency by using the Harvey's apparatus. So they all use the thermal stimuli for measuring the thermal pain behavior. But if you want to measure the mechanical pain behavior in animals, you can use the worm-free hair test or the Rental-Silito test. And there's another kind of, test by which you can measure the pain because of chemical stimuli. One is the Formalin test, and other is the acetic acid-induced writhing. So in case of hot plate test, as you know that, we are using the high temperature in this case to actually cause the pain in animals, right? But at the same time, you want to make sure that the temperature is not too high, which will damage the skin of the animals. Otherwise, what will happen? All the receptors, nociceptors, they'll be burned. So there is no purpose of doing anything there. So you want to make sure that the optimum temperature is optimum enough to cause the pain in animals. And the responses are jumping, withdrawal of the paws, and licking of the paws. So this is the time where you should remove the animals from the hot plate. Okay, and if the drug is having analgesic activity, so this test is used for the centrally acting analgesic like opioid-like drug, right? However, the peripheral analgesic like aspirin or phenylacetic acid, they do not affect this response because this hot plate response is centrally mediated in nature. So only central analgesic can show the activity in this model. So this is a general procedure. I will not go into the details. You can have the mice or the rats, depending upon the rationale of your study, and have at least 5 to 8 animals in your group. And you can divide the animals into different groups, like standard analgesic drug, which is currently in the clinic, and different doses of your test molecule, and the control groups, like healthy control or the disease control. I hope you are aware about these different controls, in pharmacological studies. And the temperature of the hot plate is around 55 degree plus minus 1 degree Celsius. And then you measure the time taken by the animal to show the response, either licking or jumping, and we record it. We call it reaction time. So if the reaction time is increased, we consider the drug is having an analgesic activity. Okay, and you can do it before the drug treatment and at different time points after the drug treatment, like 30, 60, and 90 minutes after the administration of different drugs or vehicle. Then the prolongation of latency time indicates that whether the drug is having analgesic activity or not. And based on the data, you can also calculate the ED50, which is effective dose 50. The dose which is required to produce the 50% of the analgesia. So there is another model, which is a tail flick model. Okay, so this model also uses the thermal radiation, in which we expose the tail of the animal with the thermal radiation, and we observe the withdrawal or the flicking of the tail. This model is also centrally mediated pain behavior. And morphine-like drugs, they are capable of prolonging the reaction time in this model. Okay. So here you can use either star rats or the mice and divide the animals into different groups. Okay. And then you can adjust the temperature or the intensity of the radiating source. And generally we keep the cutoff time, and the withdrawal time is almost 2 to 10 seconds. So, and the tail flick latency is recorded before and after the administration of the standard or the test compound. So these are just general ways of measuring the pain behavior in animals. So then you can calculate the ED50 values as well. Okay. So this was all about the thermal stimuli. But if you have to measure the mechanical in animals, for this we have worm-free hair test, which we use in our lab here. And you can also use the electronic worm-free anesthesiometer, which is having a digital meter where you can see how much force is there when the animal is withdrawing the paw or showing the vocalization kind of behavior. So these are the worm-free hairs with different forces ranging from 0.2 gram up to 15 gram. And you apply these here, just for example in this picture, right, the animals are sitting on a wire mesh platform, and these animals are neuropathic in nature actually. Okay, you have ligated their sciatic nerve, and when you are poking them with different hairs of different force intensities, and then you measure whether animals are withdrawing their paw, not withdrawing, not because of just movement, withdrawing, abrupt withdrawal basically, which is a clear-cut kind of sign of pain behavior. Okay, so this is a just kind of touching pain behavior, I would say, and that's why we call it for mechanical allodynia. We use it for mechanical allodynia. But there's another kind of mechanical pain behavior, which we also call a Rendal-Silito test, because this was given by the scientist Rendal-Silito. Here, you use a kind of clip basically, where you are pinching the paws of the animals between two clips. So this is actually less noxious stimuli. So if the animals are suffering from neuropathic pain or any other kind of pain disorder, they will show more pain or the decreased force, to this response. And we call it mechanical hyperalgesia. Okay. Now, this is the other kind of, pain behavior, which is because of certain kind of chemicals, so which is one of the most widely used tests, I would say, formalin pain. And this is very easy to do. You don't need any kind of special equipment, to perform this test in your lab. You just need formalin, which is very easily available in any of the lab. And you just need a video camera to record the pain behavior of the animals. And you can actually count, you can score the pain in animals. Okay. So in this case, you can have either the male Wistar rats, or you can also have the male Swiss albino mice. And you use the 0.1 ml, 100 microliter of 1% formalin, and you inject it in the dorsal portion of the hind paw, of the front paw actually. Okay. And then the test drug is administered simultaneously, either by the subcutaneous route or the oral route. It depends upon the objective of your study. What will be the route of drug administration? Okay. And the readings are taken at 30 and 60 minutes or at 120 minutes, depending upon the kinetic of the drug. Okay. And then you measure the pain behavior. Okay. Which can be tested easily by elevation or favoring of the paw or excessive licking and biting. Okay. Then you can calculate the ED50 value depending upon how many doses you have used in your test. Okay. So this test generally used to identify the central active drugs, whereas peripheral active drugs, they do not show any pharmacological effect in this test. So this is the last test which I'm going to discuss today, but it doesn't mean there are not other tests. There are many more other tests for the pain behavior, but just because of the lack of time, I'm wrapping up my talk here. So this is the acetic acid induced writhing, which is used for the assessment of peripheral analgesic activity, because you want to distinguish between whether this particular molecule is having a central analgesic activity or the peripheral analgesic activity, right? So in this case, we use the acetic acid and we inject it into the peritoneal cavity of the mice in the stomach region actually, and you observe the animals for characteristics stretching kind of behavior. You can see in this picture actually, here. So you can see in the bottommost figure actually, the animals are touching their abdomen onto the floor. Why? Because they are feeling kind of I would say the peripheral pain in their peritoneal cavity because of this formalin. So you can use mice or rats depending upon the objective of your study again, and you divide them into different groups, standard, test, and control group, and then you inject the 0.1 ml of the acetic acid, which is 1% volume by volume, in the interperitoneal cavity of the mice, and then place them in the jar for observation. And you observe for the onset of the writhing, means when the first time animal has shown the writhing, and the number and duration of the abdominal contractions for a period of 10 minutes. So if your drug is having an analgesic activity, this first thing, the onset of writhing will be delayed. And second, the number of the writhes also will decrease, and the duration of abdominal contraction will also decrease. Okay. And then you can, give your test and the standard drug 15 and 30 minutes prior to the acetic acid administration, again, depending upon the pharmacokinetic of your molecule. Okay. And then you can calculate, the writhing period and compare with the control group. And this is the formula to calculate the percent protection or percent inhibition, which is MC means mean of rise produced in the control group, and MT means mean of rise produced in the test group. This is how you can calculate, how much percent protection your molecule can offer in this model of peripheral, pain. So I would like to end my talk here. So I would like to, just end this with a brief quote, anyone who stops learning is old, whether at 20 or 80. So anyone who keeps learning stays young. So keep on learning. Learning should never end. And this is the beautiful heart of Varanasi. If you get a time to visit Varanasi, do come to IIT BHU and visit this beautiful place. Thank you.
Thank you, sir. Thanks a lot for your wonderful session. Your passion and extensive experience in pain research was very much evident in your research. It has been an honor to have you, sir, today. We felt very privileged to have you, sir, today. If anyone is having any kind of query, he or she can post their message in the chat box. We will unmute you. Anyone, please? Or you can raise your hand.
Either the talk was very clear or nothing was clear.
So it was really very clear. It was a really wonderful session and you have discussed this very nicely.
Thank you. It was my privilege to be here.
Now it's time to felicitate Dr. Vinod. Sir, kindly please accept our token of gratitude.
Thank you so much.
Thank you, sir. Thank you.
I appreciate that.
Thank you, delegates. We will meet at 1:20 sharp. There is a break of 10 minutes. We will meet again at 1:20 sharp.
On exploring multidisciplinary avenues in the pharmaceutical education and research. It's privileged to have Dr. Randeep Singh Bhaiya with us. Welcome you, sir.
Hi, Arvinder. Welcome you again on the third day of FDP on exploring multidisciplinary avenues in the pharmaceutical education and research. It's privileged to have Dr. Randir Singh Dhaya with us today. I welcome you, sir. Dr. Randir Singh Dhaya is Associate Professor in the Department of Pharmacology, School of Health Sciences, Central University of Bathinda, Punjab, Bathinda. He has a total 17 years of research and teaching experience in his credit. His research area is diabetes and the diabetic complications, such as diabetic nephropathy and neuropathy, as well as obesity. He has received a grant of worth 24 lakhs on the project entitled Pharmacological Investigations of Some Indian Herbal Medicinal Plants in the Treatment of Diabetes and its Complications from the Department of Health and Technology, DST, Government of India under SERB Fast Track Scheme for Young Scientists, FAST. He has also received a grant of rates about 20 lakhs from the Central Council of Research in the Homeopathy Department of Ayush, Government of India 2000 fellow for the project entitled to evaluate the attenuated potential of Gymnesa sylvestra, Cephalandra indica, and their bioactive compounds in the diabetic complications. He is a member of Association of Pharmacy Teachers of India, Association of Clinical Pharmacists of India, Indian Hospital Pharmacists Association, and Association of Pharmacy Professionals. He fetched Saroj V. N. Sharma Award for the best research paper from the Indian Pharmacological Society, India in 2011. He got APP Best Faculty Awards in 2018 during the 5th Indo-Australian Conference, MAPPS 2018 at College of Pharmacy, University Mulana, Ambala, Haryana. He is in top 2% most cited scientist in the world. That is the research base of Stanford University database. He got University Research Fellowship, SRF, from the Punjabi University, Patiala during his PhD, as well as AICTE Fellowship during his M.Pharmacy. He is working in collaboration with National Botanical Research Institute, Lucknow, Uttarakhand Ayurveda College, Dehradun, Department of Farm Sciences, Kurukshetra University, Chitkara University, Punjab, as well as UIPS, Punjab University, Chandigarh. He has total 110 publication to his credit. He has guided 10 PhD as well as 12 M.Pharmacy students. He has attended total 11 university level conferences, 12 national conferences, and 25 international conferences. He also organized various conferences, workshops, as well as seminars. I welcome you, sir, for today's third technical session. The sir is going to speak on the topic alternative to animals in the pharmaceutical research. Please, sir.
Thank you, Dr. Praneeth, for the introduction. And I'm very delighted to share my opinion in your FDP. And as was told that this FDP is attended by the person from the several sections, the scholars, the academicians, as well as you can say other people from the pharma field. So in this FDP, I'm going to take a very common topic, means that nowadays everybody is talking about this evaluation or the drug evaluation. But what are the options or how we can go for the drug evaluation? Generally, when we talk about the pharmacological evaluation of any drug, any compounds, so what we talk about is that we can use or we can evaluate the efficacy or the effect of any drug on the animals. But when we are working on the animals, means that simple laboratory animals just like rat, mice, rabbit, guinea pigs, so there are several issues which are being, you can say, asked that why you are using the animals, or how much is the number of animals, or is there any other alternative we can use except these animals. So I'm going to discuss my topic on this. What are the alternatives to the animal use for the pharma research? So I will share my presentation also, so that we will have some impression of how the things are different if we are talking about other alternatives to the animal use. Let me share my presentation.
Okay, sir.
So is it visible?
Yes, sir.
Okay. So this is going to be my topic. What are the alternatives to the animal use? Generally, when we talk about the animals, so animals are being used into the biomedical research, and these animals, why we are using the animals? Because what we say is that when we are using the animals, so the physiology, or you can say the body anatomical structure of the animal, as well as physiological function of these small animals, or not small, all the animals as similar to the humans. And that's why if we want to have any new drug, new medicines, and we want to test the efficacy or the safety of that drug, so we are not directly going into the humans. That's why we are using the animals, small laboratory animals, just to evaluate their effect. It can be any side effect, it can be adverse effect, or some toxic effects. That is the major use of animals into this biomedical research, as well as some of the drugs which are already into the market, and we find that there can be some potential side effects, there can be some potential adverse effect. Just to evaluate the safety of those already existing compounds, we are also using the small animal into the research. And there is a graph into this one. If we can see that if we talk about the use of animal into the different aspects of biomedical research, so major portion is that we use the animal just to understand the body in health as well as disease. And along with this, we want to check the safety of the medicines, research and development of the medicines, and as well as you can say for environmental production, as well as the species preservations. Nowadays, one more use is that we are breeding these genetically modified animals. Means that if we want to evaluate any specific drugs which is related to the effect on the genes, so we are simply modifying these genes into the animals, and these genetically modified animals are also being used into the biomedical research. So when we are using the animals into the biomedical research, so there are certain guiding principles that we cannot, although that animals are considered very important in the biomedical use, still there are certain guiding principles, and these are called as the 4R, means replacement, reduction, refinement, and rehabilitations. Means that we are using the animals, but when we are using the animals, it should be a utmost care that why we are using the animals. Means that first R of this 4R is replacement, that if there is no other use, there is another alternative, so then we can go for that. Another R is the reductions, means that ki we can get the information, same information from a small number of animals. We can reduce down the numbers. Another one is refinement. When we are using the animals into the biomedical research, there are certain procedures in which we induce the pain, we go for the surgical procedure, and other things which are, you can say, painful or harmful to the animals. So it should be considered that we should design or devise some refined methods so that we can reduce down the pain, suffering, or distress to the animal. And earlier these were 3R. Now this 4th R, this rehabilitation, has been introduced a few years back. Means that whatever animals we are using, there should be some provision for the rehabilitation of these animals into the research. Means that you have used the animal, it should not be discarded as such. So these are the guiding principles. But when we are talking about the animal use, means that although we are saying there should be reduction, there should be replacement, but if we go with the stats or if we go with the data, the number of animals which are being used globally for the biomedical research is huge. There is a society which is called as the British Union for the Evaluation of Vivisection and Dr. Hadwin Trust, which suggests that throughout the world, there are more than 115 million vertebrates are used for the biomedical research. And the top 10 countries are UK, Japan, China, Australia, France, Canada, UK, Germany, Taiwan, and Brazil. Means these are the top using country. But if we talk about the India, there's one lab in Hyderabad. This is National Center for Laboratory Animal Sciences. This lab itself supplies near about 50,000 animals to the laboratory every year, and more than 175 institutions, academic as well as research institutes, they are using. And this is the data from this lab that 50,000 animals are being used. We know that in our area, specifically if we talk about this Punjab area, there is PAU, there is Punjab University, there is NIPER also. They are also supplying the animals. So this is the data from that lab that 50,000 animals are only supplied by this lab. And if we consider other labs, other resources, or other institutions also which are supplying the number of animals for the biomedical research, this number is also very huge. If we talk about our country, means that although we are using that number of animals, but what is the condition of these animals, how these animals are being used, or how these are being treated, it's very questionable. Means that few years back there is one society which is called as the PETA, as well as CPCSU, and was a very big case. Means that one monkey was rescued, her name was Paro, and as well as there's 36 other monkeys were rescued from the NIV. And you can say that this NIV, National Institute of Virology, is a very reputed institute. Means that when this monkey was, you can say, rescued from these labs, these were in a very pity conditions. Means that these animals were inbreed. Means that 2-3 generation of the monkeys, it was there, that you can say offspring which were born, they were also into the cages. Means the natural habitat of the monkey is, because these are wild animals, they live into the jungles or the forest or the open spaces, but these animals were caged into the, these were caged for decades. Means generations were caged over there, and as a result of which these monkeys, they were in a very, you can say, pitiful conditions. Fingers and teeth, they were not into the animals. Moreover, when these animals were rescued from the cages, now they were not able to realize what is their real habitat. They were simply spinning around the cages. Means that the animals were thinking that this is their house and they belong to this area. Means they were not aware that their natural habitat is what. And as a result of which, means that CPCSC and the PETA, they took a very consideration of this issue, as well as the Renbexi lab in 2002. It was also found that dogs were being used, but these dogs were used for the study of, you can say, dermatitis and other infectious disorders. These animals were used for skin sensitization, toxicity, and other biomedical tests. But the condition of these dogs were very pathetic. Means that these animals were itself having so many diseases because the care was not taken. Although we are talking about that there are simple principles that when we are using the animals into the biomedical test, so we should consider about the suffering of the animals, and we should try to reduce the number of the animal. We should try to rehabilitate the animals. But these rules or the regulations or these conditions were not followed anywhere. That's why there was some considerations that why we are using the animals, or what are the alternative if we want to think beyond the use of animals into the biomedical research. So considering this point, again, there were different thoughts of the people, okay, that we should use the animals or not. So there are basically three type of thoughts, or the different schools of thoughts into that. One is the purist, second one is the ethicist, and last one is the revolutionist. When the purists are believe that if we want to ensure the safety or the efficacy of the drug before going into the animals, sorry, humans, so we should test these substances into the animals. Ethicists, what they are saying is that, okay, if we are using these animals, then the number should be very less, and it should be ensured that these animals are treated humanely, means their pain, suffering, distress, it should be minimized, and proper care should be taken of these animals. And last group is the revolutionists. What they are saying is that although we are using the animals into the biomedical research, but they are not serving any purpose, means that they should be replaced with some advanced technology or the advanced techniques which are available these days. And the reason for talking about these things is that why the revolutionists were saying that we should go for some advanced technology or the techniques is that although there are certain factors or the certain disease that although we are saying that the animals are similar to the human, but still there are certain conditions or the certain uh, situations where the effect of any drug into the animals was totally different. And when these drugs were given to the animals, the effects or the you can say effect of these drugs were totally different. Means that animals sometimes these are very much sensitive to the toxins, but the human means animals may be rough or maybe resist to the specific to any toxin, but humans are more prone to this one. And there is one example of which you can say every pharma people knows about the thalidomide. Thalidomide was introduced in the market for the morning sickness, and it was considered safe into the humans, sorry, animals. But when it was given to the pregnant females, that leads to the thalidomide tragedy, as a result of which near about 10,000 offsprings they were delivered with phocomelia, means their forelimbs and hindlimbs were not there. Means although it is not the case that this drug thalidomide was not evaluated or it was not tested into the animals, reproductive toxicity as well as developmental toxicity of thalidomide was done, but still it was found to be very safe into the animals. But when it was given into the humans, that it was a tragedy, it was a havoc. Means that's why the peoples are saying that although we found the drugs are safe into the animals, but when we give these drugs to the humans, the effect is totally different. Then what is the use of using animals for the safety, efficacy, and other effects to be tested into the animals when they are not serving any purpose? And another one, Dr. Richard Klusner, he is saying that we have tested the cancer, we have treated each and every type of cancer into the humans, but we are not getting the exact or the duplicate results into the humans. Every type of cancer has been cured when we are talking about the anti-cancer activity of any substance into the animals. We are getting the positive results, but when we give these substance or these new drugs to the humans, we are not getting that result. As well as that these monoclonal antibodies and other neurotherapeutics which totally behave differently when we give into the animals, their behavior or their pattern of distribution or effect is totally different. As well as when we give into the animals, the chances may be that these monoclonal antibodies, they are producing a very 100% effect into the animals, but when we are going into the humans, we don't find that type of effect. But that is the reason why the revolutionists are saying that animals are not serving any good purpose for the biomedical research. And another one example is that we know there is a good relationship between the alcohol consumption as well as cirrhosis. But when we give alcohol to the animals, we are not able to develop the cirrhosis or the liver diseases into the animals. Only the baboons In case of baboons, we can find the result, but the data which is produced into the baboons is not so much consistent. And that is the reason that although we can use any type of animals, but the pathological conditions, what we are finding into the human, as well as the pharmacological effects which are into the animals, are not replicated or not mimicked into the humans. And that is the reason why the people are saying that we should have some advanced or the alternatives to the animal use. So considering these factors, so there are different opinions, just like we have some advantages of the animals use and we have some disadvantages of the animals use. So means that if we want to go for the alternative to the animals use, so we need some skilled manpower and time consuming factors and all these things. Still, we have to opt out for the alternative to the animals. So if we talk about the alternative to animals use, we have different techniques, and these techniques are these days called as little bit more refined or technologically advanced. And these are cell culture, human testing, human tissues, in chemical testing, organ in a chip, microdosing, volunteer studies, population research, as well as computer simulation. So these are some of the alternatives which we can use, that if we simply skip out that portion, considering this that animals are not serving so much good purpose into the biomedical research. So these are some of the alternatives which can be considered for the evaluation of any new compound, any new drug before going into the human consumption. So first one is this is called as the cell culture techniques, and this cell culture is, you can say, a very good technique, basically in vitro technique in which cell and tissue these are cultured, and these cell and tissues are grown in a culture medium, and from that we develop a colony, and that colony is a single colony which can be used, and we can get the cell or the tissue from, you can say, any specific cell line also. And moreover, we can also find out the enzymes, receptor, anything we want to study our effects. So basically this is in vitro culture techniques, or considering the animals use, that if we want to evaluate our compound onto animals, so that will be a little bit costly affair. But in case of this cell culture, this is less time consuming, and we can say less expensive also, and we can use these cells, because if anything, any new compound which is affecting or which is producing toxicity into the cell, that will be toxic for the human use also, or for the animals use also. So simply by finding the cytotoxicity, we can say that this will be considered a much cheaper effect as compared to the use of animals. So means this is how we are using the cells into this one. Simply dissociate the cell from any source, and after that cultivation, we have the colony, and this colony is, you can say, any potential substance is incubated into this one. And after that, if there is any cytotoxicity into the cells, means we find that this substance is having some toxic effect, so we will not go further for the animal use or into the human use. And considering these points, so there are certain tests, just like what I were talking about, we can go with the cytotoxicity. Toxicity, this is cell viability test. Means that simple specific number of cells you can have, and after that you can add the substance or any substance which you want to evaluate. Pharmacological or toxicology of the substance can be find out. And based upon this, what is the application of this one? Means that earlier, if we talk about the toxicity studies, that animal toxicity, it can be acute toxicity, it can be chronic toxicity. We are using the different type of animals. For example, that if we want to go for the skin sensitization studies, we are using the rabbits, dermal sensitivity, and the eye irritation test on also. For eye irritation also, we are using the rabbits and the ox also, as well as for dermal sensitivity, we are using the rabbits as well as the guinea pigs. And if we think about this one, the procedure is little bit brutal in this one. Means that the any substance. It can be any chemical substance which can produce the irritation to the animals. So if we give this substance into the eye of the animal, it is causing a lot of suffering or pain to the animals. But just to skip out that thing, that procedure, we can have these tests. For example, this hen egg test on the chorioallantoic membrane. So this is means that we are taking the fertilized chicken eggs just to check the irritation of this chorioallantoic membranes. Means that if we find that there is irritability, means that there is disturbance or disruption into this skin, so definitely this substance will produce some irritation into the eyes. Means कि वो humans के लिए भी harmful है, this is harmful for the animals also. Simply कि red blood cells means कि that hemolysis is there. Simply if the hemolysis of the RBC is there, protein denaturation is there, so means we can find out that definitely this substance will have some adverse effect, or you can say deleterious effect into the humans. And this BCOP, this is one of the important tests that which is now considered by the FDA also. Earlier, when we were using this test, means the ocular dermal irritation test, that the skin of the animals were exposed. Skin of the animals were exposed to the irritating substance. As a result of which, suppose that if any substance which is highly acidic or either basic, and when this substance is being administered or it is being applied on the skin, it will definitely produce a lot of irritation. And the procedure was itself very painful to the animals also. And just to remove that one, now this BCOP test is you can say is accepted by all the industry. Even the FDA is saying that if no need to go for the dermal test, we can simply go with the BCOP test, because simply we can find out if there is positive effect into the BCOP, so this substance is safe for the human use. And these are some of the, you can say that how we are doing the procedure and all. Means that by simply replacing the animals with the cell culture technique is much more beneficial. Simply you can say that less expensive also, and you can say uh, the suffering of the animals is also reduced in this case. And as I told you that there are, you can say, some advantages. You can get the cells or the tissues from any source, and moreover you can grow the cell and tissue into your labs. And another condition is that if you are using the cell and the tissues, there are no moral or the legal issues, because when you are using the whole animals, in that case you have to take the permission from the IAC, and as well as if you want to use the larger animals, then you need to get the permission from the CPCSCA. Means that in this case there is no moral, ethical or any legal issues, because you can grow the cells into your lab, you can use the cells for the evaluation of different activities. But there are some disadvantages also. Means that the effect, because this effect is produced on the cell itself, but when we use the whole animals, in that case the effect is integrated, because cells make the tissue make the tissue system, organs and organ system also, because cell is the basic unit. So if anything is affecting the cells, so definitely this effect will be propagated onto the whole animal level also. So from this cell-based assay, we cannot predict out that this cytotoxicity effect, which how much will be the extent or the intensity of this cytotoxic effect into the whole animal. So this is the, you can say, limitation. But suppose that you want to evaluate the anti-cancer effect. So in case of anti-cancer activity, there is no need to go for the whole animal. Simply you can take the any cell lines, you can grow the cell lines, you can find out the cytotoxicity of your compound, and that is all. That is all. No need to go for these animals at all. Another one option or another one alternative is your microorganisms. You can say that we are having several microorganisms, and these microorganisms are very extensively studied. Means that we know that how to grow and what is their genetic makeup, and how this microorganism is that we can modify, genetically modify it also. So as a result of which, if you want to study, for example, this Saccharomyces cerevisiae, this is one of the most extensively studied organism, as well as E. coli is our, you can say, the very common name into the biomedical research. The genetic makeup or the genome of this E. coli is very well studied. So means that if anything we want to study it about the cell regulator, or we want to find out that if this gene is involved, or you want to evaluate your compound for any activity, for example, that in case of neurodegenerative diseases, for example, Parkinson, Huntington disease, are the effect of the drug on any protein, any gene, because we know that these genes or these proteins are present into these organisms. So simply that by finding the effect on that gene or then protein, we can find out the effect of our compound. No need to take the whole animal into this thing. But there are some disadvantages, means that when you want to use these microorganisms, you need some sophisticated instruments, as well as professional skills, a little bit time consuming. Moreover, in this case, you need a biological committee. Safety committee is required in this case. Reason behind is that some of these strains are pathological in nature, which can lead to some other disorders. So that's why these are some of the, you can say, disadvantages associated with the microorganism. Otherwise, the genome map of microorganism is well studied. Simply finding out the effect on these genes on the protein can be a very good tool to study this neurodegenerative disease. Next alternative is human tissues. Means sometimes we can have this healthy or the disease tissues from the humans as well as from the animals. Means that sources can be like surgery, isolated perfused organ, perfused frozen tissue, as well as the tissue after the postmortem. And now one more thing is that this is called as the reconstituted human epidermis. Means this is a skin model. Basically, this is commercially available also. If you want to evaluate the irritation of any chemical onto the skin or onto the surface of any chemical or the cosmetics, so then this reconstituted human epidermis is skin model is used, and this is also accepted by the FDA also. And what is done in this case, this is how we are having this reconstituted human skin. So this is just like this one, that this is our skin. So all the structure, or you can say the structural makeup of reconstituted human epidermis is same. Means that simply that you no need of animal use, you don't need have anything else. Simply that earlier how we are doing this dermal irritation test, that we use the rabbit skin into this one, that skin of the rabbit is exposed, means the hairs are removed from that, and we apply the chemical substances, as if this chemical substances is producing some irritation. So this is painful to the animals. Moreover, advantages is that this is giving the more accurate effect as compared to the rabbit skin, because we are using the human skin. So the structural permeability or the structural changes what your chemical substances will produce into the human use will be the same into this case. That's why we can skip out the rabbit for this model. We can simply go with reconstituted human epidermis skin model. Next are the in chemical tests, and now most of the labs are going with this one, is that if you are having some sophisticated instrumentation, you can go with the HPLC training and all. For example, that some shellfish mixture, there is one concentration of the shellfish, that if it is going beyond than 30 microgram per ml, means that is toxic. So if you are having any sample and you find, you simply run a test into the HPLC, and you find from the HPLC you can easily find out the area under curve. From the area under curve you can find out the concentration, and you find that the concentration is more than 30 microgram per ml. So means that will be toxic. So there is no need to going for any human test or any animal test. By simply chemical technique, you have found, okay, this substance is toxic. So we are not going with this one. Next one is organ on a chip. This is a little bit advanced also, and we are very much hopeful for this technique to be developed. Actually, organ on a chip is a multi-channel 3D microfluidic cell culture, and this is integrated cell culture. If you can see this one, this is just like human structure. That this heart is there, bone, kidney, liver, gut, all these things are there. Means ki that is itself is an integrated system. Means ki that there are tiny chamber, whole chambers are there. Means ki that the drugs, when it is going inside the humans, inside any body, living organism. So this is the pattern. Drug is for absorption, distribution, metabolism, and the excretion. Similarly, it is passing through the different organs. So what is happening into this organ? For example, if we are giving the drug and is moving into the liver, so what is the metabolism? What will be the effect of the liver onto that drug or that substance? This will be evaluated. Means simply this is circulating in that way. We can find out that if this is going into the animals, but why we are using the animals? Just for the pharmacokinetic as well as the pharmacodynamic effect. So means that we can find out the pharmacodynamic effect or the pharmacokinetic effect into this method also. And considering this factor nowadays, because the technique is under transition phase, so we are having different organs nowadays that lung on a chip, gut on a chip, means all these organs are available. So you want to evaluate the effect of any drug onto the kidney. Simply that you will have a kidney model on a chip and you give your compound and you will find all the data that what can be the possible effect of your compound onto the kidney or any other organs by using this one. Means कि that we can, these are some of the applications. Drug metabolism आपको मिल जाएगा. You can exactly mimic the condition into the lung, kidney, heart, as well as gut. And but we are having, you can say, the whole structure human on a chip also, because these organs are integrated with each other. But there are some limitations as compared to the human animal use, because some of, you can say, less information is provided. And sometimes when we are using this one organ on a chip, means these are the small capillaries or small channels are there, and sometimes that if any air bubble or the trapping of any substance is there, so this can disrupt the overall flow, as a result of which it can damage the cells and you will get some different type of effects as compared into the normal conditions. Next one model is whole cell simulations. Means that sometimes we can easily we can find out the effect into the whole cell. Means that structures of the cell, means cell, what are the organelles present into this one. This is being developed and we can simply give up the drug and we can simulate simulate the cells, that if these substances being given into the cells, so what can be the behavior, physiological behavior of the cells. Another one method is our microdosing studies. What we are talking about, then we give drugs to the animals just to find out their potential toxic effect into the humans. But if we simply go with the microdosing means that if I want to find out the pharmacokinetic of any substance, means that pharmacokinetic means we can go with microdosing, means that very small doses of potential new substances, means that उसका metabolism कैसे होगा, absorption कैसे होगा, distribution like this way. So means microdosing can be done, and this microdose can be labeled also, and their pattern, the distribution pattern can be easily analyzed by the mass spectrometer. Means that why we are using this model microdosing, but generally what we are saying that the ADME of animals as well as humans is same. But you can say 40% of the drugs, they fail the drug trials just because of the pharmacokinetic patterns. means at ADME के basis पे 40% clinical trials are failed. So if we can simply go with this method, microdosing studies, that very small dose, which is not having any potential toxic effect, is given into the human, and we can simply find out the pattern of the ADME into the human itself. So there is no prediction, just like what we are doing in case of animals. We will give the dose to the animals and we will predict that similar effects can be there. Means still there are some chances of error or some chances of difference. But when you are giving this type of microdosing to the animals, because the pattern of the drug will be same, ADME will be the same, so we can have some very potential exact effect into these conditions. Another one model is this one, volunteer studies, because sometimes that you can have some volunteer. For example, if you want to check some if drugs or some compounds which are having some nutritional benefits, which can be used for the drug addiction also and pain also, because what we are saying that in case of pain or drug addiction or nutrition, we cannot find out the exact behavior into the humans. So that's why in this type of studies we can have some volunteer studies. But these volunteer studies should be considered in that case that these are not producing any harmful or the painful effects into the humans. Another one, this is, you can say, a little bit more common or more advanced also, and nowadays everybody is talking about the in silico studies or the computer aided drug designing. And as a result of these advanced techniques, a large chunk of time is being saved. What are these in silico studies? Basically, in silico is a Latin word when silicon chips into the computer. And this is one of the very important alternative to the animal studies. Means that in this case, we can design the drugs, we can go for the ADME, or simply that on the computer, you can easily find out that we will design a molecule with this type of structure. This molecule will combine on these receptors, and these receptors are present over there. So these can be the potential therapeutic effects. This can be the potential side effects of the drug. So basically, drug discovery process, the time of drug discovery process is significantly reduced by using this in silico studies. So there are different type of in silico studies also. We can say bacterial sequencing techniques, means that we can take the DNA or RNA of this bacteria and we can make the sequence of these RNA or DNA by using polymerase chain reactions. And these bacterial DNA or RNA, their proteins can be evaluated, key that if any drug is affecting that these protein, RNA or DNA, what can be their potential effects into the humans, or what can be their potential therapeutic effects. And there are different in silico models also. We can say that computer aided molecular drug designing, quantitative structure activity relationship, computer assisted learning, computer or the mathematical analysis. In case of molecular modeling, means that this is how the drugs are designed and how they are reacting with the receptor. For example, that this Trevor Marshall, this has used that if we are using this some structure just like vitamin D metabolites, because they can how they will produce their effect onto the receptors, and whatever predictions are we are getting into the molecular modeling, because we know that drugs and receptors, they are having complementary structures. If you know the structure of any receptor, so you can design the drug accordingly so that it will bind onto that receptor and it will produce the effects. And that's what we are doing in case of molecular modeling studies. And this is, you can say, computer aided drug designing. If we say that we can simply go with this procedure, that we can predict a molecule, we can identify a molecule, we can design a drug as per the structure of the molecule, and this is how you will lead. Generally, if we talk about the process of drug discovery, it takes near about 10 to 12 years for any molecule to become a drug. And if we talk about the cost of that procedure, this is near about $5,000 million for one molecule to become a drug. Time as well as money is involved, because what we are going through the preclinical studies after the clinical studies, because, and what can be the results after this 10 to 15 years and $5,000 million, the molecule can fail finally. But if we are going with this procedure that you are simply designing the drug as per that you can say receptor, and you are validating that target, and after that target you can say other lead optimization, and from that you can find out preclinical as well as the clinical, and moreover you can predict out that this can be the potential therapeutic effect, this can be the potential side effects or the adverse effects. So means the process is simply the time frame of drug discovery is simply reduced by this process. Moreover, this is not so costly. If we talk about the process into $500,000 million, and this one that you are having a software of 10 to 12 lakhs, and you are simply predicting, you are designing the molecule, you are putting this molecule on the receptor side, and you are finding out the effects. So more potential where you find less bonding energy is there, means that this substance is having more potential for this type of receptor. So we will use this substance for this disease. Means that is what is being done in case of computer-aided drug designing. So as I told that it is very cost saving also, and it will speed up the process of drug discovery. Simply on the computer you can design the drug molecule, you can evaluate their effect, you can find out their potential benefit of any new compound. Next one is quantitative structure activity relationship. I think that every pharma person is quite aware of this thing. What is the meaning of quantitative structure activity relationship? When any drug is binding to the receptor, it will produce an effect. But if we can change the structure, means that suppose that some substrates are being changed, so we can find out some different effects. So the QSR softwares are also there, and that is the reason behind although we are not having that much new molecules into the market, but just changing the structure, we can find some better effects by changing the lipophilicity of the drug, by changing the hydrophilicity of the drug. You can change the some effect, you can reduce some side effects of the drug. So this is what we are doing in case of quantitative structure activity relationship, and this is how we are doing all these models in case of QSR studies. So means that QSR studies are having some advantage that you can easily find out the relationship between the structure activity, as you can say physiochemical properties, and based upon this interaction you can design the drug accordingly, that this type of drug will have more pharmacological or more significant effect as compared to the other drugs. Another technique which is being used is computer assisted learning. Means that computer assisted learning is basically a platform which is providing learning as well as it is providing the information. Means that there is a basically an interactive platform which produce or which provide education. Nowadays one software is there, up to date software, means that the drug information is there, and this one stopcat, this is very commonly used. This is toxicity prediction by computer assisted technology. Means that before introducing any substance into the market or before the preclinical also, we should have some toxicological profile of that substance. For that we want to go for the acute oral toxicity, skin irritation, dermal toxicity, and all these things. So but topcat, what it is doing is that it will simply predict that this substance, if you are going to use into the dermal toxicity or eye irritation toxicity, these type of effects will be produced. So there is no need to go for this process also. Another example is that gypmer, means that we can say that I think few years back this CPCSA has banned the use of animals for graduate courses for the demonstration purpose also. As a result of which, this JIPMER has developed a software when the animal experimentation is there. You can say X pharmacology is one of that software. Means all the experiments, because कि आपको देखना है कि that what is the effect into the epilepsy or locomotor test. All the information is there. All the procedure from the computer assisted learning. You can simply find out that if you are giving this type of drug, you will find this type of effects. Means simply that if you are using a heart or the tissue into that, you are adding the drug, what where will there be any contraction or the relaxation? All the things are there. No need to find out the tissues, no need to sacrifice the animal, take the tissue from that. And if you want to evaluate some just for the demonstration purpose, you want to go for the anti-epileptic activity, you don't need animals into that. Okay? Simply from the software, you can easily see that if we are giving any new compound, or what can be the effect, or how the epilepsy will be produced into the animals. So this is nowadays considered very significant contribution of this in silico methods. Next method is your epidemiological studies. These epidemiological studies are, you can say, I will say this is a pool of data. Means these studies are being conducted for several years. Means that sometimes there is one studies which were conducted for 21 years. And what it was done that if this pattern is there, means any disease is there, any population is there, there are changes into the living pattern, or there is any changes into the eating pattern, lifestyle pattern, what can be the consequences of that factor onto the human health? Or if you are adding something into this one, for example, that we know that smoking is associated with the lung cancer. And how it was found, that it was found from this epidemiological studies. If any person who is smoking for that much period, so this means that it will lead to some sort of cancer. And this is the study which I was talking about, Framingham Heart Study. It was conducted for 60 years, and now it is also ongoing. And from these studies means that several factors related to these disease, means prevention, symptoms, and evidences, these were found. For example, कि उसमें से हमें that how this obesity is related, and how the puberty is affected in case of fat women. And for example, that sleep apnea is responsible for the increased risk of stroke. Means that from this type of epidemiological studies, we can easily find out that these factors are related with any disease or any pattern. Whereas that, and these studies are really considered very significant into the research. So that was all from my side, that how we can have alternative for the animals into the biomedical research. And I would be very happy to answer any queries from any side.
Thank you, sir. Thanks a lot. It was really a very wonderful session. You have discussed very nicely about the alternatives to the animals used in the research, such as cell culture, cell viability test, human tissue culture, in silico studies, in chemical testing, computer simulations, etc. Really, it was a very informative session. Now the session is open for the queries. If anyone is having any kind of query, he or she can post their message in the chat box. We will unmute you. Anyone from the audience? Okay, sir. I think no queries there. Your session was really very informative as well as clear. Thank you very much, sir. Now it's time to felicitate Dr. Thaya. Sir, kindly accept our token of gratitude. Thank you, sir. Thank you very much.
Thank you. Thank you very much, Dr. Praneeth.
Now, I invite Ms. Jasveer Kaur, Assistant Professor in the Pharmaceutics, for the formal vote of thanks. Ma'am, please.
Good afternoon, dear all. Today, I just feel incredibly honored and privileged to have been allowed to extend vote of thanks. On behalf of GHG Khalsa College of Pharmacy, Sudhar, the entire organizing team, and on my own behalf, I extend a very hearty vote of thanks to eminent speakers for today's session, Dr. Sunita Garg, Dr. Gurpreet Kaur, Dr. Vinod Tiwari, and Dr. Randhir Singh Dhaya for gracing their important work and sharing with us their findings and opinions. I would like to thank the entire management of GHG Khalsa College of Pharmacy, Sudhar, for support and encouragement. A special thanks due to Principal Dr. Satvinder Kaur for day-to-day support in this faculty development program. Once again, I would like to propose a vote of thanks to all the speakers and the participants for becoming part of today's session. Thank you all. We will meet on 12th August, and I want to wish you all happy Raksha Bandhan, and we will meet on 12th August. The schedule for the FTP will be shared on WhatsApp. Thank you all. Have a great day.